A big hello to all my wonderful PU2 aspirants. How are you all, children? Please do not be worried about biology. We are going to ace the biology examination. I just expect 100% effort from your end, and I'll also give my 100%. So are you ready? I hope you're ready. So let's have this positive affirmation. I know that biology is too much amount of theory, and there are a lot of things to remember. There are not so many people who are good at remembering things. I get it. So it's always better to do things in the format of questions. It gives you more memory power. So what you do is, whenever you read your NCERT, make sure you're reading a chapter within 15 to 30 minutes, not going beyond that. Because if you give more time, you will try to utilize that time. Reading does not take too much of your time. You get it? Then when you solve questions, you will be understanding a lot of concepts. And I've already given you part 1 of top 80 plus questions, where I have discussed 4 chapters, that is sexual reproduction in flowering plants, human reproduction, a very small chutku chapter, that is reproductive health, and also evolution. So 4 very important chapters are being done there. Please go watch these videos, my dear children. Remember, when I say top 80 plus questions and things like that, where am I getting these questions from? I am getting these questions from the question bank that the government has released this year, the revised question bank. So which is why I want you to solve these questions. You get it? This might be easy, might be difficult, we never know. Let's solve it together. In that way, understand the concepts as well. Then ma'am, what about the 2 marker, 3 marker, 5 marker? Those questions that I did were the previous year questions and the model question paper questions. Okay, so you have to give equal weightage to all these things. Okay, I know it's too much to ask for, but I have given you perfect strategy. Please follow it, and please, whenever you have your NCERT, okay, whenever you have your NCERT, here also I have segregated the chapters, right? So what you can do, after reading one particular chapter, watch the 2 marker, 3 marker. Section wise I have divided. For example, I have read sexual reproduction in flowering plants. I go watch the 2 marker, 3 marker, all the 2 marker, 3 marker questions of sexual reproduction. Then I go to 5 marker questions. I read all that. Then I come over here. Then I read the sexual reproduction questions over here as well. So in that way, I am covering what PYQs as well as question bank. And in that way, I am understanding the concepts and also the type of questions that are coming. You get it? Now, you might feel this is a big task. So once you start doing it, you will feel fine. Trust me, you will feel fine. And you might see so many same questions here and there. You're like, oh, I know this. Okay, so please not be worried. And in this particular session, I am going to take 4 very important chapters. One is 2 very important chapters actually. One is molecular base of inheritance and principles of inheritance, and then inheritance and variation, and then you have 2 chutku chapters, that is, I think I chose ecosystem and biodiversity and conservation. I think so, yes. Okay, so I want you to put your attention into it, and let's start off this particular chapter, that is principles of inheritance and variation. Let's give it a target of 40 minutes. Let's see if we can complete all the questions in 40 minutes, okay? So what is test cross? Write its significance. 40 minutes for this chapter. Okay, what is test cross? What do you mean by test cross? For example, I have a tall plant. I do not know whether it is tall like this, capital T, or it is tall like this. What do you call this condition as? This is homozygous tall and this is heterozygous tall. Hetero as in they are different, but still it is dominant over this, which is why it also appears tall. Now, ma'am, why do I need to know all that thing? Basically, whenever I want a pure breed plant, I want a plant like this. So to make sure whether the plant is actually tall or not, I do a test cross. What do you do in a test cross, ma'am? In a test cross, we are going to cross the individual with a homozygous recessive parent. Recessive individual as in, it's like this. It will be like this. So what is the use of this? Let me show you. So over here in your NCERT, there is an example being given. So this is the recessive parent. Okay, which is white color flower. Here also you have recessive parent, that is white color flower. Now you want to find out whether this violet flower which is there, it is violet like this, that is WW, capital W, or is it violet like heterozygous? So when you cross this, okay, when you cross this and this, what do you get? All the progeny, my dear children, will be violet in color, because all of it will be having the dominant one dominant allele over here. So all of them are going to be violet. Whereas when you see over here, when this fertilizes, when this fuses with this, you get capital W, small w. Okay, here you get capital W, small w. That means both of them are violet. What about them? They are small w, small w. Small w means it is white in color. So here you get a combination of violet as well as white flowers. That means this particular violet flower that you're seeing is like this. It is not a pure breed. So I cannot be utilizing it for any crop breeding purposes, because if I do, I might get the recessive trait which I do not want. You get it? So I am targeting for this particular thing, and that is what you mean by test cross. A test cross is a cross between an individual with an unknown genotype. I do not know the genotype. This is the genetic makeup. Genetic makeup is known as genotype. Okay, and homozygous recessive individuals. It helps determine whether the unknown genotype is homozygous dominant or heterozygous. Significance is what? Determine the genotype. Helps identify whether an organism is purebred or hybrid. It's a purebred or a hybrid. Hybrid as in it is heterozygous, or purebred as in it is homozygous. Now it is used in breeding, helps breeders select desirable traits in plants and animals. Very important point, my dear children. Now listen, you might feel what is the importance of all these questions that I am putting over here. Trust me, on the day of examination, if the concepts come and you already know it, you get an immense sense of happiness, and I am not here to waste any bit of your time. Remember that. Even I am putting my 100% effort, standing here for 4 hours for recording videos straight. It is very difficult, you know. In class and all, when you talk, there is a communication between you and me. We both talk, but here it's us just talking to you guys. So it needs a lot of energy, and if you're putting that energy, that means it is very much important. I hope you get it, yeah? So get things clear with question number 2. A pea plant with purple flowers. Imagine, okay? A pea plant with purple flowers are crossed with plant having white flowers. The progeny produced only purple flowers. Only purple flowers, my dear children. Again, if it is only purple flowers, then probably, if capital P stands for purple, then one parent could have been like this, another parent would have been like this. Then only all their progeny would be what? Purple in color. Both the parents should be homozygous. Now on selfing, the plants produce 482 plants with purple flowers, 162 plants with white flowers. Explain the mechanism that counts for these results. So first of all, you have to tell that capital P stands for dominant allele, which is for purple, and small p stands for what? Recessive allele, which is what? White, my dear children. Now what do? You do the parent cross. You do parents over here, right? Parents over here. Put capital P, small p, then put gametes. Here the gametes are what? Capital P, and small p, right? Now you fuse the F1 generation. What do you get? All of them, when you try to fuse, it is capital P, small p, this also capital P, small p, this also capital P, small p, this also capital P, small p. Aren't all of them purple, my dear children? Yes. So F1 generation is all purple. What is the mechanism behind this? Basically, there is a dominant allele, and this is stated in law of dominance. That is where F1 generation shows only purple flowers, since P is dominant. Capital P is dominant over what? The small p, which is white. Now the second mechanism is where you are selfing. That means in between the progeny, you are making them cross. So what happens when you cross them? So it is capital P, small p, and capital P, small p. You are doing selfing. When you do selfing, what do you get? These are the gametes. These are the gametes, agreed? This and this fuses, you get capital P. This and this fuses, you get capital P, small p. This and this, you get capital P, small p. This and this, you get capital P. You get small p, small p. At the end, you get, where am I? Small p, small p. So if you see, there are 2 capital P, that means it is going to be purple in color. There are 2, again, there is 1 capital P over here, so this is going to be dominant. So these are also going to be purple. How many purple? Basically 3 of them are going to be purple. 1 of them is going to be white in color. Now, over here they have mentioned that it produced 482 plants with what? Purple flowers. And then 162 plants with what? White flowers. That means total is what? Total will be, I think it's 644. It is what? 644. Okay. Now, here we saw 3 is purple and 1 is what? White. Let's see if it is matching over here. So, 3 by 4 into 644, what do you get? You will be getting 482. Now, 1 by 4 into 644, what will you get? You will be getting 162. That means this is correctly matching over here. What is the mechanism behind this? Basically, the mechanism behind this is the law of segregation. What does law of segregation says? That is, whenever the gametes are being formed, my dear children, from a pair of alleles, only 1 allele will be going to each gamete. Only 1 allele. So this is a pair of chromosomes that we have. This is capital P, this is small p, suppose. Both the chromosomes are not going to go. Half of the number of chromosomes are going to go. So either this can go or this can go. If this goes, it is capital P. If this goes, it is small p. If both of them went, my dear children, this wouldn't have been possible. This thing wouldn't have been possible. So on selfing, The F2 generation follows a 3:1 phenotypic ratio, producing 482 flowered and 162 white flowered plants, confirming what? Mendel's law of segregation, which says that each organism carries 2 alleles for a trait. Agree? 2 alleles for a trait, right? Now, which separate during gamete formation, ensuring that each gamete receives only 1 allele. Yes, each gamete which is there, this is a gamete, it receives only 1 allele of the pair. You get it? So this is the mechanism that accounts for this particular cell. So talk about Mendel's law of dominance and also talk about Mendel's law of segregation. Please show this cross and explain your answer. You do not have to write big paragraphs. Make sure you're writing little, but make sure you're writing parent, gametes, F1 generation, and then again parent, gametes, F2 generation and all, and write the phenotypic ratio. How do they appear to us? That is phenotypic ratio, physical thing, right? So 3 of them are purple. I can see it, ma'am. I do not know the genotype, but 3 of them are purple and 1 of them will be white. Okay, and they exactly follow this. Fine. Now going ahead to the next question. Yeah, I have written all of this thing. If you want, you can go ahead. So it's not, okay, it's 2. Okay, let's go ahead. So the calculation you only do, my dear children. I have not done it, okay, as of now. But anyways, it will be around this thing. If at all there is calculation mistake, do not blame me for that. Okay, actually you do not need to write all this thing. It's just for your understanding that I have given. What you have to write is over here. Everything is over here. So please do the calculation. If at all there is calculation mistake, I think it's 482 or something like that. Okay, anyways. What do we have now? Name the respective pattern of inheritance where F1 phenotype does not resemble either of the 2 parents and is in between the 2. Okay, so basically F1 generation, for example, I am crossing a red flower which is like this and a white flower which is like this. Okay, when I crossed it, the gametes are what? The gametes are this. Cross this with this, you get capital R smaller. Cross this with this, you get capital R smaller. Capital R smaller, capital R smaller. What do you expect? If this stands for red, if this stands for white, what do you expect is the color of the flowers which are over here? It should be red, but you know what? It is not red. It is pink in color. It is pink in color. And what do you call this? This is known as incomplete, this is known as incomplete dominance. Incomplete dominance. It is not completely dominant, my dear children. R is not completely dominant over this chutku small r that we have, and this is found in snapdragon flower. This is a very important example. Snapdragon flower, or you also call it antirrhinum. Okay, also you call it antirrhinum majus. That is the name of the flower. So does not resemble either of the 2 parents and is in between the 2. It will be what? Incomplete dominance. Incomplete dominance. My dear children, your math sir is disturbing me. He actually came here and is dancing in front of me. If I could show you, I could have done that. Okay. So yes, next question. Resemble only one of the 2 parents. What is it? Resemble only one of the 2 parents, as in you have capital T, and then you have small t up over here. Okay. So when you cross this, what do you get? This and this fuses, you get capital T, small t. This and this fuses, you get capital T, small t. Same thing. Capital T, small t. Capital T, small t. Either all of them here, my dear children, are they going to represent both of them together? No. It is going to represent only one parent. That is, all of them over here are tall. All of them are tall. Why is this being shown? Because this is totally dominant over the other one. So I'll say this is what? Dominance. When only one of the 2 parents are basically visible over here, that means it is showing dominance. It does not resemble either of the 2 parents, I will say it is incomplete dominance. Works? Okay, please remember snapdragon, the name of the plant, and also antirrhinum majus, that is the scientific name. Chalo, next. Yeah, this is the answer. Now, what are multiple alleles? Give an example. Multiple alleles as in, ma'am, until now we just studied that, for example, if you're talking about height, it is capital T, small t. But ma'am, in some things, okay, in some characters, there are more than 2 alleles. There are more than 2 alleles. My dear children, this is known as multiple allelism. Multiple alleles refers to the presence of more than 2 alternative forms of a gene occupying this locus on a chromosome. That means over here, suppose this is a locus as in location. Over here, I say it can be IA, it can be IP, or there is one more situation where it can be IA and here it can be IO. Ma'am, how many genes, how many alleles are there? IA is one allele, IB is one allele, IO is one allele. 3 alleles are there for one particular character. What is this particular character? This is your blood group, my dear children. This is what? The blood group. We have 3 alleles for one particular gene, that is the blood group. So IA is 1, IB is 1, then IO, or you can also write it as small I. Either way, these are the 3 alleles that we have. So ABO blood group system in humans is controlled by 3 alleles. That is, this is an example of multiple allelism. Works? Chalo. Next thing, a child has a blood group O. A child will be having a blood group O. If the father has a blood group A and the mother has a blood group B, work out the genotypes of the parents and the possible genotypes of the offsprings. Okay. So blood group, okay. Fine. Over here, look at me carefully, okay. So we have the parents, the father is having A blood group, okay. The mother is having B blood group. The son, right, son or whatever it was there, the progeny which is there, at least one of them is having O blood group. How is that possible, ma'am? I'll tell you. So remember this funda that I'm gonna tell you over here. Will you be able to remember it? Let's see. So for A blood group, the person can be IA. Okay, both the alleles can be same. Then it is A blood group. Or else the person can be IO. What do you understand from this? That is, A is dominant over O, which is why still the blood group will be A only. Now talking about B blood group. Now for B blood group, again the person can be IB, or the person can be IB, IO. What do you understand from this? B also is dominant over O. B also is dominant over O. Now we have O blood group. In that case, it should be IO, otherwise there is no chance. That means you understand one thing, that it is a recessive blood group. It can only show its appearance if it is in recessive format, otherwise it won't. Now, there is one more blood group, ma'am, that is AB blood group. What happens in that? So, in AB blood group, there is IA and IB. Ma'am, both of them are expressing themselves. How can it be AB? Yes, both of them are expressing themselves, and this is known as codominance. When both, if the question was, what happens when both, what is this thing where both the parents are able to express their character? It will be codominance, my dear children. Both of them are equally dominant, which is why both of them will be expressing together. Okay. Now, in this scenario where I am saying that a child has O blood group. Now, O will be coming only when you have 2 O's. You have 2 O's. In that condition, my dear children, from both the parents, you will be getting 1 O. You should be getting 1 O, then only this can happen. So, the father's genotype will be obviously IA and IO, and mother's genotype will be IB, IO. Now, over here, since B is dominant, mother will be having B blood group. Since A is dominant, father will be having A blood group. Now, if you find, these are the parents' genotype. Okay, now if you talk about the gametes, what do we have? So, IA and then IO is the gamete. Over here, IB and IO is the gamete. When you try to fuse, so it is IA, IB, this is IA, IO, and this will be IB, IO, and this will be IO, IO. Now, over here, see my dear children, okay? This is B, this is A, this is B if you do not understand my writing, and this is O. So, over here, one child has IO, that means this is the child that has O blood group. Now, you are asked about the different genotypes of other progeny. So, genotype of the progeny can be IA, IB, IA, IO, and IB, IO. So, this is a progeny having AB blood group, this is a progeny having A blood group, this is a progeny having B blood group. So, please check your blood group also. Talk to your parents and check their blood group, and then you can find yours and your siblings' blood group as well. Okay, what is the probable thing? You'll understand it better. Okay, will you be able to write this? It's easy, no? It's fun. Okay, now, where are we? Yes. So, father with blood group A can have IA or IB, this is the same thing. Therefore, the genotypes of the parents must be IA, I. Small I means IO, okay? And here also the same thing. If you do the Punnett square, you will be finding like this. I told you these are the genotypes that you have. IA, IB, IA, IO, and IB, IO, and then II, that is O. Okay, this is the Punnett square. You can make in the form of Punnett square as well. Chalo, next. Define pleiotropy, give an example. What do you mean by pleiotropy, my dear children? What do you mean by pleiotropy? Pleiotropy is when a single gene Okay, usually a single gene is responsible for one particular trait, but over here a single gene is responsible for multiple traits, not just one particular trait. So a basic example of this is phenylketonuria. Have you heard about the disease? It's a genetic disorder, okay? And what happens over here, the gene which is going to form a very important enzyme, that is phenylalanine hydroxylase, that is mutated. Okay, that is mutated. So why do we need this enzyme? Because in our body we have phenylalanine, which is an amino acid. It has to be converted into tyrosine. It has to be converted into tyrosine, and this is done by the enzyme that is phenylalanine hydroxylase. So suppose the gene coding for phenylalanine hydroxylase is affected. Do you think this transformation can take place? No, it cannot take place, and hence when it is not happening, phenylalanine is going to accumulate in our body in the form of phenylpyruvic acid, and it is going to cause mental retardation. It is going to cause mental retardation. Now if I tell you, this is not the only thing that happens. This is one trait. There is one more trait that this particular gene handles. Since phenylalanine hydroxylase is not being formed, tyrosine is not being formed, and you know tyrosine is a very important thing needed for the formation of melanin, which is responsible for our pigmentation, for our coloring. Now if melanin is not there, what will happen? We will be having reduced pigmentation in our hair and in our skin. Our hair and our skin, my dear children, will have reduced pigmentation. So mental retardation is one trait. Over here, reduced pigmentation is the next trait handled by the same gene, which codes for what? Phenylalanine hydroxylase. So pleiotropy occurs when a single gene influences multiple unrelated phenotypic traits. They are not what's related, right? They are unrelated phenotypic traits. Example is phenylketonuria. Please write like this: is a genetic disorder caused by a mutation in the gene coding for the enzyme phenylalanine hydroxylase, leading to what? Accumulation of phenylalanine in the brain, which causes mental retardation and reduced tyrosine, which is there, my dear children. It results in decreased melanin production, leading to lighter hair and skin. It is reduced pigmentation as well. Will you forget this? Please not forget this. And there is one more term that is known as polygeny. That is known as polygeny. I am explaining you here itself. What do you mean by polygeny? Here you said that one gene is coding for multiple traits, right? Multiple traits. Then I am saying that multiple genes, ulta, okay? Multiple genes are coding for one trait. This is polygeny. That means you have multiple genes, my dear children. Didn't understand? I'll tell you. So example is your skin color. Example is your skin color. Your skin color is handled by so many genes. So suppose this is a chromosome, this is another chromosome, this is another chromosome. So over here you have capital A, capital A, capital B, capital B, capital C, capital C. This is going to give out the most darkest skin color. This is going to give out most darkest skin color. So we see gene 1, gene 2, gene 3 all are responsible for skin color. This is the darkest one. So your your genotypic genotype will be capital A, capital A, capital B, capital B, capital C, capital C. Okay, this is the genotype. Now again, suppose there is change over here. This is small a. Suppose this is small a. Then what will happen? So over here the genotype will be capital A, small a, capital B, capital B, capital C, capital C. This will be lighter than this guy. This will be lighter skin tone than this guy. Now again, suppose this is also small b, this is also small b, and this is also small c. Again you are getting more lighter skin tone, my dear children. Now you might ask, ma'am, what is that? How do we get the lightest skin tone? How do you get the lightest skin tone when all of them are recessive, my dear children? So this B is small b, this C is also small c. So it will be A, small b, small b, small c, small c. This is the lightest skin tone. This is a very important MCQ question. Which one will give the darkest skin tone? This will be this. Which one is the lightest skin tone? Wherever you find more and more recessive alleles, that means this one will be giving the lightest one. Okay, done, clear? Chalo. So that is polygeny. Now going ahead, list 2 features of Drosophila melanogaster which makes it suitable for genetic experiments. So T.H. Morgan, please remember T.H. Morgan was the person who used the fruit flies, or what do you call it as, Drosophila melanogaster, for his experiments proving what? Chromosomal theory of inheritance. So chromosomal theory of inheritance was put down by Theodore Boveri and Sutton. Please listen to me carefully, okay? Chromosomal theory of inheritance was put down by Theodore Boveri and Sutton, right? Sutton and Boveri, that's what we say. So over here, one very important thing is that What do you mean by chromosomal theory of inheritance? It says that whatever behavior is there in the chromosome, same thing is exhibited by the genes. That means probably the chromosomes over here might be carrying these genes that we are talking about. Now, to prove this and all, you had T.H. Morgan coming into action, and he said that I am going to use this guy for my experiments. So, what are those features that you definitely need in Drosophila melanogaster for the experiment? So, why did he actually use Drosophila melanogaster? That is the actual question. So, it can be grown on simple synthetic media. It does not say that I'll be growing. It's not like I do not want so many fruits. I can grow in simple synthetic media. Okay. Now, it completes the life cycle in 2 weeks. So, if at all we want to see any changes, we can observe within 2 weeks itself. We do not have to wait for years. Now, a single mating produces a large number of progeny. Again, I get more chance to experiment on a lot of other progenies which are coming. Okay. Now, clear differentiation of sexes is there, my dear children. Male and female is easily distinguishable. You can make them mate. Now, many hereditary variations visible under low power microscope also. What else do you need? So, you're asked about 2 things. I have given you 5 points. Either of them, if it comes in a 3-mark question, you can answer it still. Okay. Will you be able to remember it? You should be. Now, define mutation. What are mutations? Mutation causing agents known as. First things, one very important thing, my dear children, if you think my pace is very much, okay, it's too difficult to understand, I'll request you to go to the settings, okay, you can lower down the pace. I have increased the pace because otherwise the video will go very long, and there might be children who are just here for revision, not explanation. I'm still explaining for the children who are not understanding. That's why I'll request you one thing from your end. I want, that is, if you feel the speed is way too much, please decrease it a bit. Okay, in the setting column you can decrease the speed. Now, what do you mean by mutation? Mutation is any sudden change that happens in the DNA sequence or in the genetic material, my dear children. Okay, and this change is heritable. You can basically transfer this to the next organism. For example, I have a change in my DNA, and when I have my babies, what will happen? Basically, they are also going to get this particular copy of what I am having. You get it? So, this is a heritable change. So, mutation is what? Sudden heritable change that occurs in the DNA sequence. Now, what do you mean by mutagen? Mutagens are the agents that cause this mutations. Okay, the agents that cause this mutation, what do you call them? Mutagens. For example, carcinogens. Okay, they are certain things that causes cancer. Cancer is a form of mutation, right? So, basically, such like that, you have what? It's not a form of mutation, it's a result of mutation. So, yes, mutagens are the agents that causes what? Mutations. Works. So, next thing. Schematically represent a pedigree chart of myotonic dystrophy. Okay, this question was there in the question bank. So, myotonic dystrophy is given nowhere, but in the pedigree, if you have seen this, just after the pedigree analysis paragraph, they have given a picture like this, okay, where they have mentioned in below this that this is the chart of myotonic dystrophy. Myotonic dystrophy is an autosomal dominant trait, my dear children. It is an autosomal dominant trait. That means even if one organism has this like this, for example, these are the 2 genes. I have A and A. Suppose this at least one is affected, okay, one is affected, that means it will be showing up that person will be diseased. That person will be diseased. That is what you mean by autosomal dominant trait. Even if one particular thing is there, it will express itself. That person will be diseased. Over here, if it's like this, then definitely you do not have any option. But this also can be a reason for a person to be diseased. And whenever you see dominant trait, my dear children, I will give you that example also. You might be asked to find what kind of pedigree is this. So, always remember, if it is a dominant trait, it will appear in every generation. Okay, for example, this is the parental generation, here you observe it, okay, then now in the progeny generation also you observed it, now when they mated in their progeny also you observe it. So, in every generation you see, do not go like this is not affected, then how can they be affected? Here it is not visible, but the affected ones will definitely pass it down to their generations. You get it? That is how you find the dominant traits. And autosomal, how do you find it? It's not sex linked, it is just autosomal. Autosomal as in, it's not on the sex chromosome, it is just on the normal other chromosomes which are there. Okay, it does not have to do anything with the male and the female, everyone can get it. So, here as you can see, here is the female who is getting the disease, here it is a male. So, there is no clear differentiation for that. So, it will be an autosomal dominant trait, my dear children. This is for myotonic dystrophy. This is for myotonic dystrophy. So, whenever it's not a hard fast thing to remember everything, just that make one parent affected, okay, then make 2 children affected, then their children also at least one should be affected. If you make like that, then also it's fine. Do not go around making all of them affected, okay, please do not do that, that will be not correct at some point. Okay. Now, this is also one thing that is being asked. So, whenever I find a relatable thing that could be asked, I have putting it over here so that you don't miss out any information. Now, this thing is the usual thing that is asked. What do you mean by this? In MCQ, this is asked. What do you mean by this? This is the male, this is the female, but it's the mating between relatives, my dear children, and it is known as consanguineous mating. What do you call it? Consanguineous mating. It's a mating between relatives. That's why you have two lines. They're closely linked. Okay. Now, this is just mating. Whenever it is colored, that means it is affected. Whenever it's mentioned like this, that means 5 unaffected offsprings are there, and not mentioning the sex of the organism which is there. Okay. What do you mean by this? That the parents with the male child affected with the disease. Parents with the male child are affected with the disease. So, this means that the male child is affected with the disease. Okay, I got confused. So, these are things, this is not usually asked. This is the thing that is being asked to make sure you know about it. Okay? Okay, cool. Let's go ahead. 10th question. Schematically represent a pedigree chart for sickle cell anemia. Now, sickle cell anemia, what type of disease is sickle cell anemia, my dear children? Try to recall. Before I tell the answer, you should be recalling it. So, it is autosomal, it is not sex linked. Okay, it is definitely autosomal. But is it recessive, dominant? What is it? It is a recessive disorder. Autosomal recessive disorder, my dear children. Okay, so that means recessive means it can skip generation. It can skip generation. Recessive means that it will only appear. It will only appear if both the parents have the alleles. Not if only one parent have it. That means not both the parents have the allele. Basically, would be appearing when both the alleles are there, defected alleles are there. That's when it gets shown up. Okay, otherwise it won't be shown up. So over here, suppose this guy had HBA and HBS. This is the affected allele. This is the affected allele. This is the normal allele. So this guy would not be having the disease. This guy would not be showing up the disease. This guy will be just a carrier. That is this. Okay. Now same with the female also. She also will be just a carrier. Now suppose in the progeny at least once this and this will come together, my dear children. This and this might come together. That's when you say that they are disease. So the parents can be carrier. This is why I say that they can skip generation. You won't find them in every generation. So they are skipping generation over here because they are just the carriers. You're passing it to other organisms. Again, these guys also have it, but they are not showing they are carriers. You can find it the next generation. So whenever you are making a recessive trait, make sure you are skipping the generations which are there. Okay, the parents might not be affected. In the simpler sense, you can remember like that. The parents might not be affected, but the progenies might be affected. Okay, and this is autosomal again. Why? Because there is no sex linked. It is not sex linked. Okay, all the organisms are getting the disease irrespective of the sex. So it is autosomal recessive disorder. This is sickle cell anemia. Next. Distinguish between alpha thalassemia and beta thalassemia. So, alpha thalassemia and beta thalassemia, my dear children, are different forms of thalassemia. What is thalassemia? It is again an autosomal recessive disorder. It is again an autosomal recessive disorder. So, what happens over here is that for alpha thalassemia, you have HBA1 gene, okay, and you also have HBA2 gene. Here also you have allele for the same thing only, so I will write HBA1. Here also you have alleles for the same thing, that is HBA2. So how many genes do you have? You have 2 genes and both the genes have 2 alleles. Both the genes have 2 alleles. That means for alpha, basically there are 4 alleles. There are 4 alleles and 2 genes. 2 genes and 4 alleles. Okay. Now talking about beta thalassemia, I'll tell you what I am trying to say. Okay. This is HBB. And this is also HBB. So it is just having 1 gene, and 1 gene has 2 alleles. 1 gene has 2 alleles. For beta, there is 1 gene. Sorry, I'll write 2 alleles first and then I'll write 1 gene. Now listen, these genes that you see over here are going to code for alpha globin chain. You know, in hemoglobin is made up of alpha globin and beta globin chain. These genes are going to code for alpha globin chain. Suppose there is a mutation in this. Suppose there is a mutation in these alleles, my dear children, what will happen? The formation of alpha globin will be affected. Similarly, this is for beta globin chain. If there is a mutation up over here, what will happen? The formation of beta globin chain is affected. At the end, if alpha or beta, anything is affected, what is going to be affected in overall sense? It is the hemoglobin molecule, which is why I'll say it is a blood disorder. It is what? A blood disorder. Where do I find these genes on which chromosome, my dear children? This is on chromosome number 16 and this one is on chromosome number 11. Now suppose over here, this is affected and this is affected. That means still alpha globin will be produced, but it is not produced that much. It is not produced that much. Suppose all of them are affected, that means alpha globin chain is not produced at all. So I will call this as a quantitative disorder. That means I am going to mess around the quantity. If 2 alleles are affected, that means they are still being found, but the amount of alpha globin chain which is being produced, it is less. Here also, when I say that this is affected, that means beta globin chain is produced, but the amount is what? Really less. So I will call this as quantitative disorder, not qualitative. So please remember, thalassemia is a quantitative disorder. The previous thing that we just studied, that was sickle cell anemia, it is a qualitative disorder. Here you are going to mess around the quality of the beta globin that is produced. So what happens is that there is a different mutation over here. This is also same gene, HBB gene. So there is a different mutation happening up over here, which is why the quality of beta globin which is being produced, it is bad. It is what? Bad. When it is bad, the hemoglobin which is formed from this is also going to be in a bad state. And then hemoglobin is carried by RBC. This RBC is also going to be in a bad state. So usually we have a normal bioconcave disc shaped RBC, but here since it is carrying a deformed hemoglobin, the cell's shape will be sickle cell. It will be what? Sickle cell shape. I hope you understand that. So mutations are different happening over here and there. Here the mutation is happening because there was a codon, that is GAG, which is now replaced. Over here the A thing is which is there in the mRNA of the gene that codes for beta globin, my dear children, there it should have been GAG, but instead of GAG it is GUG. A is replaced by U. So GAG was responsible for the formation of glutamic acid, and I hope you know that the mRNA is the one that carries all these codons. It is responsible for formation of so many amino acids. These amino acids comes together to form a protein. What is the protein over here? Beta globin. So over here, this one amino acid is being changed in a chain of amino acids to what? To valine. And because of this chain, there's a deformed beta globin that we get, and the deformed beta globin will obviously make this hemoglobin also mad, and hence the RBC would be what? Sickle shaped. So this is on chromosome number 11, same story, but different type of mutation is happening over there. This is an example of point mutation. At one particular point, mutation is happening. Only at one particular point, which is going to change everything. So how do I remember it? A comes before U, so A was there before and then it is replaced by U. So G comes before V, so the glutamic acid was there before and now you have valine. So this is the sixth codon. Sixth codon as in you might be having, for example, A, B, 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 oh not B, 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 sorry, C, 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 G, 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 and then T, T, sorry, not T, 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 U, 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 1, 2, 3, 4, and then 5, A, T, no, A, U, A, and then one more codon, that is G, A, G. So 1, 2, 3, 4, 5, this is whole mRNA, my dear children. On the sixth codon, this thing is happening. So I have explained you sickle cell anemia also over here. Okay, so this is difference between sickle cell anemia and thalassemia. That is a qualitative disorder. Quality is getting disoriented. Here the quantity is getting disoriented. So alpha thalassemia is caused by mutations or deletions in HbA1 gene or HbA2 genes. Okay, these genes are located on chromosome number 16. Severity depends on the number of gene deletions. Number of gene deletions as in this or this or this, how many. Okay, it is a quantitative disorder. Now caused by the mutations in HBB gene. Okay, HBB gene, you can write like that also. This gene is located on chromosome number 11. Severity depends on whether one or both the alleles are affected. That's all. Okay, both of them are autosomal recessive disorder. Both of them are blood disorders. So if you are asked about blood disorders, qualitative and quantitative, just give example of both of them. Fine. Now, write the genotype of an individual who is carrier of sickle cell anemia gene but apparently is unaffected. So, carrier of sickle cell anemia would be what? The normal is, my dear children, the normal is HbA and HbA. You write like that. But the person who has sickle cell anemia, Individual who is a carrier, he is not having the disease, he is a carrier. So he or she, whatever it is. So it is HBA and HBS. So this means that this is a carrier. So there is a defective allele, we can pass it to the next organism. And individual affected with the sickle cell anemia disease, then it should be HBS and HBS. Which is why, you know what, previously it was said that we should not be marrying our cousins or our relatives. Why? Because we would be having these genes. We would be having these genes. So if at all marriage and everything happens, these genes can combine together. Okay, you get it? So that is why it is totally said that you should not be marrying the relatives. I hope you get it. Okay, chalo, next thing. Study the figures given below and answer the question. Identify in which of the crosses the strength of linkage between the genes is higher. Give reasons in support of your answer. So there is a gene given over here, there is a thing given over here. You have to tell that which of them has high amount of linkage. First thing, what do you mean by high amount of linkage? If at all the genes are closely located, you say that they have high amount of linkage. So here I see the yellow and the white which is there. The genes for coding for yellow and white, they are very close to each other, my dear children. Whereas when you talk about white and miniature, they are kind of very far from each other. They kind of very far from each other. So whenever, suppose it's like this. So there is yellow, and there is white, and there is something else over here. There is something else over here. So what happens is that, you know, the chromosomes, they recombine, they kind of do crossover with each other. So even if crossing over happens, my dear children, you might be observing something that is, suppose this and this part got exchanged. Still yellow and white would be what? Close to each other. Why? Why? Because they are tightly linked. Because they are tightly linked. So I will say the one that is tightly linked, shows what? Less amount of recombination. Shows less amount of recombination. This was said by T.H. Morgan only. He only studied this. Okay, now there was a student of him who was known as Alpha Stewart. He said that the distance between the genes, it matters. Okay, so if the genes are located far away from each other, they will be showing less amount of recombination. Okay, sorry, they will be showing more amount of recombination. If the gene distance is less, that means they are going to show less amount of recombination. Suppose now the same scenario, if it was like this, like happening over here. So there is white and there is miniature. Okay, and this is a chromosome which looks like this. So if I change the parts, okay, if I exchange the parts after recombination, what do I get? So over here I will be having this one, and here you will be having the white one, and here you will be having the exchanged part. So if you see the exchange, my dear children, initially both of them were together, but now you have them on different. So recombination is what? Higher. Why? Because the linkage is less. They are loosely linked. There is too much distance, which is why there is more amount of recombination that is possible. And I'll give you easy formula to remember it, something that you can relate. So you and your best friend, if you're very close to each other, there is no chance there is another best friend that can come in. So you're tightly linked. That means what? Less amount of recombination, new friends, less amount of new friends, recombination. Recombination means something new, right? So less amount of recombination is there. Now you and your friend are not really close to each other. There are higher chances that this friend can make another best friend also. So they are loosely linked. That means they can form more best friends. So it is more new combinations that can be happening. So you can remember like that. It's an easy way to remember. Now they can give you that 37.3% recombination is seen and 1% recombination is seen. So 37.3 recombination means it is way too much. It is way too much. That means the genes which you're talking about will be loosely linked, because 50% is the maximum amount of recombination. If in that 37.3 is mentioned, that means it is what? Loosely linked, there is high amount of recombination. If 1.3 is mentioned, that means it is tightly linked, they show less amount of recombination. Okay, all the questions related to this I have explained it to you. Please remember the name of the student. Please go and read the NCERT. In linkage, the name of the student, what he said is given. Okay, he was responsible for production of gene mapping, discovery of gene mapping, and T.H. Morgan said all these things. Okay, going ahead. So you can go through the reading once if you want. Now explain sex determination in the case of human beings. So I'm going to make it really fast. So already 40 minutes are done. Oh my god, I'm so sorry children, but yes. Okay, now over here XX is there and XY is there. Okay, XX means it is a mother. It is a mother. XY means it is a father. Okay, now the mother will get two type of gametes, X and X. The father will be given X and Y. Okay, when they combine, you get what? This combines with this, you get XX, this combines with this, you get XY, and if you see, my dear children, there are 50% chance for the child to be a female because it is XX, 50% of chance is there the child can be a son. Over here, in human beings, you saw something that is known as male heterogamety. Male heterogamety, what do you mean by that? Males are producing two type of gametes. One is carrying X, other is carrying Y. Here the females are producing they're producing same type of gametes, that is X and X. You get it? So this is female homogamety, this is male heterogamety, which is being seen. Now, probability, there are 50% probability of it to be son and 50% probability for it to be a daughter as well. You can go through all this thing. I have already explained everything. Okay, nothing is left. So humans have 46 number of chromosomes, including one pair of sex chromosomes, that is this and this. Male heterogamety, male produces this. That 50% of sperm carry X, 50% of them carry Y. Female homogamety, they produce only one type of egg, all over carrying X chromosome. And when they cross, you get all this thing. Please make sure you are making this, otherwise you are getting unda. Okay. Now, sex determination in Drosophila melanogaster. Have I not explained the same thing? Okay. In Drosophila melanogaster also, it's the same thing. Now, you might feel, ma'am, it's just an insect. Yes, it is an insect, but it is having XX, XY type of sex determination. Drosophila melanogaster has XX, XY type of sex determination, even though it's an insect. Insects usually have different type of sex determination. I'm going to talk about that as well. Okay, but Drosophila melanogaster has XX, XY type of sex determination. Please remember it. Now, explain sex determination in the case of birds. Birds again, my dear children, they will have ZW and ZZ. They have ZW and ZZ. You know what? ZW is female over here. So, females are the ones that are going to produce two different type of gametes. So, I will say that females are showing female heterogamety, whereas males produce one type of gametes, that is Z, that is carrying Z. So, again there is 50% of chance for it to be a male or a female. Either way, but the only difference is that they are showing female heterogamety. This is a very important thing in your NCERT. There is no other examples given for female heterogamety. This is the only thing, that is in birds. Okay, so sex determination in birds, female heterogamety points. Chromosome composition. Birds have ZZ, ZW type of sex determination. Males ZZ have 2 Z chromosomes. Female ZW have 1 Z and 1 W chromosomes. Female produce 2 type of gametes, that is this and this. Male homogamety, that means males is producing only 1 type of sperm. This carrying what? Z chromosomes. Okay, going ahead next. Yes, explain sex determination mechanism in grasshopper or I can say insects. I am talking about insects. I told you, right, Drosophila melanogaster is still an insect, but I will say it has XX, XY type of sex determination. Rest all of them, including the grasshopper guy, has a different type of sex determination. I am going to tell you what. So, over here XS is going to be a female. Okay, there is only one where the females show heterogamety, that is birds. Rest all it's same, right? I don't know why when I come to the studio, it starts itching. Yeah. Over here, you have XO. You have what? XO. What do you mean by that? So, males can have gametes that have X chromosome. Males can produce gametes that does not have any chromosome. That is what you mean by XO. So, it's like 0. They have only one X chromosome. There is no other chromosome, my dear children. So, in a gamete, there can be no sex chromosome. In a gamete, there can be X chromosome. So, suppose this and this fuses, you get XX. This and this fuses, you get XO. So, whenever there is XO, that means it is going to be what? A male. And whenever there is an XX, that is going to be a female. Again, can I say this is an example of male heterogamety? Yes. Males are producing 2 type of gametes. One that has X, another that does not have anything. Okay, please remember. So, again, grasshoppers follow the XO type of sex determination. Males XO have only 1 X chromosome and no Y chromosome. There is no Y chromosome at all. Females XS have 2 X chromosome. Male heterogamety, 50% of carry X chromosome, 50% of them have no sex chromosome. Okay, explain with diagram. Please ensure that you are talking about parents, then gametes, then fertilization, and also the zygotes which are being formed. That is F1 generation. Okay, please mention like this. Draw it properly. You are gonna do really good. Next thing, explain sex determination mechanism in honeybees. Now, that is totally different. Okay, sex determination mechanism in honeybees, it's kind of different. What is happening over here? So, female is having 32 number of chromosome. Male is having 16 number of chromosome. Okay, in normal cell, female is having 32 and male is having 16. How, ma'am? Basically, what happens is that the females will undergo meiosis. the females will undergo meiosis for the production of gametes. Remember, for the formation of gametes, you need meiosis to happen usually, right? So they will undergo meiosis, and at the end they will get cells having 16 number of chromosomes. The males instead, they already have 16 only. They already had 16 only, so they did not undergo meiosis. So they undergo mitosis. They just increase the number of cells. So they also have 16 over here. So please remember, males undergo mitosis, females undergo meiosis. Okay. Now suppose the female gamete fuses with the male gamete. 16 plus 16, 32. It is going to be a female. Suppose this particular gamete which is there is not undergoing fertilization. So this is, as such, it is being what? A male, my dear children. And what do you call this process as? This is known as parthenogenesis. Remember, parthenocarpy is different. I always say that this is parthenogenesis, where without fertilization, male is being formed over here. This is known as haplodiploid sex determination. Haplodiploid as in what? 32 means it is diploid. 2n number of chromosomes is there. Half of it is haploid. So haploid one will be always male. Diploid one will always be females. Okay, so sex determination in honeybees, that is haplodiploid system, females, queen and workers. Okay, so females which can be there, it can be forming of queen or a worker. Usually there is only workers being formed. For queen to be formed, you have to give royal jelly and things like that. That does not happen so easily. Okay, so they are diploid, having 32 number of chromosomes, developed from fertilized eggs. So females are developed from fertilized eggs. Males, that is drones, they are just used for copulation, nothing else. So there is a flight, nuptial flight you call it, when the queen bee will be flying up. So all the drones which are there, they will also fly, they will fight with each other to copulate with the queen. So their function is just to copulate. So they are haploid and they develop from unfertilized egg through parthenogenesis. Chromosome based sex determination. The number of chromosome sets determine the sex. Diploid, that means female. Haploid, that means male. Unique feature, sperm is produced by mitosis instead of meiosis, right? The sperm is produced by mitosis. Now males have no father and cannot have sons. Yes, males, do they have father? Can they have sons? Can they ever have sons? No, the males can only come with a female this thing and produce a female only. Male can never produce what? A son. You get it? Male can never produce a son. It's a female egg that can convert into what? A male thing. So it is that. So males have no father and cannot have sons. So there is a very important line which I am forgetting, that they have grandfather but not something like that. So please go check that particular statement as a very important thing for the MCQ. Okay? In haplodiploid thing, you will be finding it. Cool, let's go ahead. Now, the following pedigree shows a particular trait which is absent in the parent but found in subsequent generation irrespective of the sexes. Analyze the pedigree and draw a conclusion. What? So, following pedigree shows a particular trait which is absent in the parents. In the parents it is absent, but is found in subsequent generation irrespective of the sexes. Oh my god, sexes. Okay, analyze the pedigree and draw a conclusion. I told you, when both the parents are not showing it, but then the progeny it is showing, that means it is what? Autosomal disease and it is what? A recessive disorder. It is a recessive disorder. When both the recessive genes come together, that's when they show up the disease. So parents can be a carrier, but over here the progenies will be having that disease. And since over here the male and female, either of them are affected, I'll call this as autosomal recessive disorder, my dear children. But how do you conclude it? So trait is absent in parents but present in offspring. That means this indicates that trait is inherited as in a recessive manner. In a recessive manner. Second thing, trait appears in both males and females equally. Since both sexes are affected, the trait is likely to be autosomal rather than sex-linked. If it is sex-linked, then there are chances that the males are affected more and things like that. Now, pattern suggests autosomal recessive inheritance. Parents are likely carriers, that is heterozygous for recessive allele. Affected individuals inherit two recessive alleles, that's when they have the disease. So I told you the parents can be like this, HBA, HBS. Here also HBA, HBS. So this is a carrier sickle cell anemia, this is also carrier for sickle cell anemia. When they join, you get one thing at least which will be having HBS, and that will be the diseased one. Okay, so conclusion is that trait follows autosomal recessive inheritance where unaffected carrier parents can pass the condition to their offspring. Give the structure answered, you will get good marks. So this type of questions also come. So you might think, ma'am, are you thankful for this or not? Tell me that. So I spotted these questions because I thought that these questions could give you a heart attack on the day of examination, which you definitely do not want. So even the question is easy, my dear children, since it is different pattern, you might not get it. But I am telling you, you might face some other questions also. On the day of examination, what do? Do not lose your calm, my dear children. Just stay there and think, okay, I studied this, I know this, and this is how it's linked, and that is how it goes. Okay, now identify. I know I do too much of drama at times, but it is good for you and also good for entertainment. You might be tired of studying, right? Yes. So identify the genetic disorders given below. Sterile female chromosomal complement 45 with XO. What is this condition? So my dear children, if you know, there are 3 chromosomal disorders. Chromosomal disorders means chromosomal disorders are formed when you have more or less chromosome. Usually we have 46 number of chromosomes. Do you know that? We have 46 number of chromosomes. 44 of them are autosomes, and then we have 2 X chromosomes. 2 sex chromosomes, either XY or XX. So 44, 45, 46 in total we have 46 number of chromosomes. Okay? Now listen to me carefully. Chromosomal disorders is a result of aneuploidy. Aneuploidy means gain or lose of electrons. So either you can gain extra chromosome or you can lose an extra chromosome, and this is known as aneuploidy. Very important thing. Discussed in 2 marker, 3 marker, 5 marker. You can go watch that. Now over here I am saying the female is sterile and the chromosome complement is what? 44 with XO, that 45 with XO. So probably 44 autosomes are there, but it should have been XX, but it is XO, my dear. And that means there is no sex chromosome happening over here. There is only 1. That is why 44 plus 1, 45. What is this condition? This is the condition of Turner's syndrome. Turner's syndrome, where, my dear children, it should be, so one parent gave the X chromosome, another parent didn't give any chromosome, my dear children, which is why this condition arises. This is when it should be a female, but it's not a female. So this is a sterile female who would be having masculine body and everything. They will be having, why they are sterile? Because they have rudimentary ovary. That means ovary is all not well developed, because they do not have X chromosome to success that, right? So basically their ovary is not well developed. They are not going to produce eggs, which is why they are known as sterile females. This is Turner's syndrome. Now again I'll take this one first and then go with the second one. So sterile males gynecomastia karyotype of 47. What do you mean by that? Sterile males are there and they have gynecomastia, as in they have the formation of breast. Now in males breast it's not common, right? So probably there's some problem over here, and they have 47 chromosomes as well. There is a gain of chromosome. Here there's a loss of chromosome, here there's a gain. So 44A plus the condition is XXY. So it should have been just an XY, but there's an extra X over here, leading to a condition that is what? XXY, my dear children. That means 44 chromosome, 45th, 46, 47 chromosomes are there. This is known as Klinefelter's syndrome. This is known as Klinefelter's syndrome. Okay, where the male will be sterile, they will be having certain feminine structures like gynecomastia and everything. Okay, done. This is because of gain, and this is because of loss. Now short-statured, furrowed tongue and characteristic palm crease. This is the feature of what? This is the feature of Down syndrome. Down syndrome, my dear children. What happens in Down syndrome? Okay, listen, both of them were having problem in the sex chromosomes which are there. Now this is the problem that occurs in the autosomal chromosome. So the 21st chromosome, one you get from one parent, another you get from another parent, agree? But from another parent you are getting 2 chromosomes instead of 1. So they didn't, probably what happens is that we have 2 chromosomes here, 2 chromosomes here. From this pair 1 comes, from this pair 1 comes. But it might be possibility that 2 of them are coming together, they are not separating. So at this condition, how many chromosomes do you have at 1? Chromosome number 21, how many of them you have? You have 3 of them. Instead of having 2 of them, 1 is there as an extra. So I will call this condition as trisomy. Trisomy of 21st chromosome. Trisomy of what? 21st chromosome. So what is a karyotype? This is known as karyotype. So it will be what? 45. It's now autosomes that is affected. So 45A plus XX if it's a Down syndrome person who is a female, or it can be 45A plus XY if it's a Down syndrome person who is a male. Okay, so this is trisomy, and this is a trisomy, this is monosomy. One is lost, so it is monosomy. Okay, please remember these features are really important. They can ask you what are the features of the person who is having Down syndrome, and also this. Cool. Chalo. Next. So I have explained all this thing. Now, schematically represent inheritance of color and shape of seed in the pea plant. You have to represent it like this. Very important question. Okay. So how do you exactly frame this thing? So color and shape. So my dear children, usually you find it difficult. So I'll just explain it in a very fast pace. So listen, when I'm saying it's round and yellow, I mean that we have capital R, and capital Y, agreed? Now if I say it is yellow and, sorry, not yellow, it is wrinkled, that is the shape, right? It is wrinkled and green, that is, it's the recessive thing, that is small y. Okay, how does the chromosome appear to you? The chromosomes are like this. Imagine this is capital R, capital Y. Here it is small r, small y. Now when gametes are formed, I told you how are gametes formed. From a pair, only one chromosome is going to form to come to the gametes. This is known as law of segregation, right? So from this pair, whatever you do, capital R is going to come. From this pair, whatever you do, capital Y is going to come. So this, whatever I do, I'll get 4 gametes, all of them having capital R, capital Y. Similarly, I'll get all the ones that are having small r from here and small y from here. Agreed? This is the gamete that we have. Now, F1 generation means you have to fuse this and this. So, I am going to fuse capital R, small r, capital Y, small y. How, ma'am? Basically, this is R and this is the Y that we had. This is the small r and this is the small y chromosome that we had. When you combine both of them, what do you get? You get something like this. You get something like this, where this is capital R, small r, capital Y, small y. Agree? So, this is dominant, which is why it is like this, and here also this is dominant, which is why all of them will be what? Round and yellow. All of them will be round and yellow. But is it like parent? No. Parent were pure, they were heterozygous. Now, Mendel was not sure, he wanted to do one more experiment. So, what did he do? He did selfing. He did selfing between these 2 parents. Okay? Now, let's do that. Okay, so whenever I do that, it will be going off. So, we have the parents now, capital R, small r, capital Y, small y. Similarly, capital R, small r, capital Y, small y. Okay, how do you form the gametes? If at all you are confused, make the chromosomes elsewhere and try to find the answer. So, if you are finding difficulty in forming the gametes, you can do this. It's a very helpful technique. So, it is capital R here, small r here, capital Y, small y, right? Now, from this, one can come, so I write capital R. From this, one can come, so I write capital Y. From this again, one can come, so I can write capital R. From this again, one can come, I write small y. From this one, so small r this time and capital Y this time. Small r this time and small y this time. Okay. Similarly, here also you get the same gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Just make all the combinations possible. Now, what do? Make a Punnett square. Okay. Make a Punnett square and basically put these terms. So, this capital R and then here also okay. Now, when you fuse this with this, you get capital R, capital Y, capital Y. When you fuse this with this, you get capital R, capital Y, small y. In that way, you will get so many combinations, my dear children. 16 combinations you will be getting. You will be getting 16 combinations which will be looking like this. I hope you got the gist of it. Now, I will show you the combination that is being formed. Here, it looks like this. Okay. Now, if you see among them, my dear children, again, you get a phenotypic ratio that is 9:3:3:1. That means the looks, how do they look? Physical appearance. So, 9 of them are going to be, always remember, the highest number is for the parental trait that is round and yellow. Okay. The lowest one is for the recessive trait that was what? Wrinkled and green. Now, this 3 and 3 you have to form new combination. So, round was with yellow, this time you are going to make round with what? Green. And wrinkled what's with green, this time you are going to make wrinkled with yellow. You get it? So, that is how you make it. So, 9 yellow round, 3 yellow wrinkled, 3 green round and 1 green wrinkled. Fine? Fine? So, if you are asked, okay, I will come to the next question later on. So, this is the thing that you have to make. I hope I was able to make you understand that. Going ahead with the next question that we have over here is what? A colorblind father has a daughter with normal vision. The daughter marries, okay, a colorblind father. Listen, color blindness is a sex-linked recessive disorder. Sex-linked recessive disorder. That means if both the chromosomes are affected, it will show up the disease. Now in the case of males, since they do not have two X chromosome, even if one is affected, it will show up the disease. So if I say color blind father, that means it is what? XCY. He has a daughter with normal vision. He has a daughter with normal vision. Now normal vision as in, my dear children, if the father is XC, anyways the daughter which is going to be formed will be having XC. Now if she has to have a normal vision, that means there will be a normal chromosome coming from the mother. That means she would be a carrier for sure. This particular daughter is going to be a carrier for sure. Or I'll repeat it, listen to me carefully. If I say it's color blind father, that means he is going to pass his X chromosome. You get X chromosome from father and the mother, right? And now I am saying that the father passed X chromosome. Still, if she has normal vision, then probably she has got one normal X chromosome from the mother. That is why she has normal vision. She is just a carrier. Now the daughter marries a man with normal vision. Now the daughter marries a man with normal vision. That means over here it is not affected. Okay. Now, what is the probability of a child to be colorblind? So how do we find it? We make the gametes so it can be X, XC, and then X and Y. When you fuse them, okay, fuse this and this, you get XX, that is normal female. When you fuse this and this, you get XY, that is normal male. When you fuse this with this, you get XCX, that means it again is a carrier, and this you get XCY. This is the probability of a child to have color blindness. XCY means there is no other chromosome to basically show its effect. So this guy will be affected. This guy will be affected, my dear children. Okay? To be color blind, so there is only one chance of this. So I say 50% of the boy would be color blind. Okay, so boys are how many? This many. So this and this. So I'll say half of the boys which are going to be produced, they are going to be color blind. Half of the females which are going to be produced will be carrier of color blindness. So understanding the genetics of color blindness. Color blindness is an X-linked recessive trait. Males need only one affected X chromosome to be colorblind, like that. Okay. Females need two affected X chromosome to be colorblind. Now the father is colorblind, that means it is like this. The daughter has a normal vision but must have inherited the XC chromosome from the father for sure. Since she has normal vision, she must have received a normal X chromosome from her mother. Might have received like that. Thus the daughter's genotype is what? This, she is a carrier. Now the probability of the son being colorblind is 50%. Okay, and the probability of a daughter being colorblind is also, daughter being a carrier for colorblindness is also 50%. Make sure you're making this, do not make any mistakes. Remember, if you want to know the daughter, okay, suppose the father is like this. What about the mother that gives the daughter to be a carrier? Listen, the mother can be a carrier as well. A mother can be carrier as well. Suppose this chromosome came, that's how she became normal. So we never know whether she is a carrier, but she is a carrier or not, but she will be having one X chromosome for sure, that is there normal. That's why she is passing. She is able to pass it on. Okay. Cool. I hope I was able to explain you this. Please make sure you're writing this properly on the day of exam. Now observe the dihybrid cross and answer the questions given below. Okay, write the phenotypic ratio. What is the phenotypic ratio? We just discussed. This is a normal thing that we discussed. 9:3:3:1. Identify homozygous dominant genotype in the progeny. Homozygous dominant as in when both they are like this. That is, so only one of them will be like that. Only one will be like that. So I'll say capital Y, capital R, capital R. Okay. Now identify a recessive genotype in the progeny. There is only one which is completely recessive, which is this. Small y, small r, small r. Okay. The only one that is completely recessive, only one that is completely dominant, like that. Now, number of parental type progeny, my dear children, you will be having number of parental type progeny. What do you mean by parental? They didn't mention that it has to be genotype or the phenotype that has to match. Parental type means round and yellow, and also wrinkled and green, right? Round and yellow and wrinkled and green. So basically over here, how many of them are parental like? So 9:3:3:1 we discussed. So 9 of them are round and yellow, 1 of them is wrinkled and green. So 9 plus 1, I'll say 10 of them have the parental type progeny. Now number of recombinant type progeny. Number of recombinant type progeny, as in 3 plus 3, they are the new combination that we got, that is 6. If they asked about genotype, same like parents, what is a genotype? So in that case, there is only 1 and 1. So this is the parental genotype that we started with. But that's not the question. They're just asking number of parental type progeny. So they're not talking about genotype, they're talking about phenotype. So round and yellow, wrinkled green, 10 of them. Number of recombinant type progeny, it is 6 of them. Okay. So your answer becomes to look like this. Fine. Works. Chalo. Next question. Given below is a representation of amino acid composition of relevant translated protein, a portion of beta chain of hemoglobin. Related to the shape of, okay, is this representation indicating a normal human or a sufferer from a certain related genetic disorder? Give reasons. So I told you the GAG which is there, it is having, okay, wait, 6 codon, it is glutamic acid. Is this representation indicating a normal human or a sufferer from certain related genetic? Give reasons to support your answer. So this would be normal human, right, my dear children? This would be normal human because there is no, it is still coding for glutamic acid only. If the GAG was converted to GUG and it was coding for valine, then it would have been what? Sickle cell anemia. So I don't know what is the answer for what I have written. So representation indicates a sufferer of sickle cell anemia, which is not the answer, okay? This is not the sufferer. Please, 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 please. So how do you reframe the answer? This, you say that this person is having a normal gene, okay, he or she is not a sufferer. If at all, okay, if at all there was a mutation in beta chain of where glutamic acid is replaced by valine, then it would have been what? A person who is having what? Sickle cell anemia. Sickle cell anemia. Okay, but as of now it is not, because over here as you can see it is in mRNA, it is GAG only which is coding for glutamic acid. So it's totally fine. Now, okay, what difference would be noticed in the phenotype of normal and the sufferer related to this gene? What difference would you notice, my dear children, if at all you had a difference? So normal individual is HbA has round bioconcave red blood cells, my dear children, whereas when we talk about sickle cell individual, they have crescent or sickle shaped. You remember this, okay? They have sickle shaped RBC that tend to clump together, blocking blood flow and causing pain, anemia and organ damage. Now carriers usually asymptomatic but may experience mild symptoms as well. Now, who are likely to suffer more from the defect, males or the females? My dear children, it is an autosomal recessive disorder, not a sex-linked one. So male and female equally can have the chance, right? So both males and females are equally affected as mutation is autosomal and not sex-linked. So on chromosome number 11, which is an autosome, okay, we have only chromosome number 23, which is a sex chromosome. Now over here, since both sexes have 2 copies of genes, I'll say homozygous suffer equally regardless of the gender. That's all. Only the first question change, okay. So basically over here, there is no change mentioned. If the change was mentioned, okay, if the change was mentioned, that is here if it was GUG and instead of glutamic acid it was valine, then probably I would have said that they have the disease. But here we say it is not the sufferer, it's a normal gene, but if it was like this, then this person would have sickle cell anemia. Please write that and come, because that is the question. Give reasons to support your answer. You have to support your answer, right? Fine. Next question. A cross between a normal couple resulted in a son who was hemophilic and a normal daughter. Okay. Okay. A cross between a normal couple resulted in a son who was hemophilic. Now, hemophilia, again, it's a blood disorder only, but it is a sex-linked blood disorder. It is different from sickle cell anemia and thalassemia. Okay. And here you are not talking about the mutations in beta globin chain and things like that. Hemophilia occurs because of mutation in a gene that codes for a protein, and this protein is responsible for clumping, so clotting of the blood, okay, for clotting of the blood. So one particular thing is gene is being mutated, which is why you get this particular disease where the blood flows on forever if there is a cut. Okay, so over here when I say normal couple, okay, and I say that the son is hemophilic. The son is hemophilic and there is a normal daughter as well. So how do we frame this? Son is hemophilic. Okay, this is XX and XY. Okay, for the son to be hemophilic, I'll say mother might have been a carrier, because both of them are normal couples. Both of them are normal couples. If this guy had H, then probably he would have had the disease, but he does not have the disease. That is why I am saying that the normal couple, so XX and XY, so normal couple, with that we say this is a person who has what, say, hemophilia. This is the carrier for hemophilia. She is the carrier for hemophilia. I'll write it properly. I don't know what happened immediately. Okay, so this is XH. Cool. Now let's make the gamete. So gamete can be X or XH or X and Y. Now this and this fuses, you get X and X. This and this fuses, you get X and Y. This and this fuses, you get X and XH, which is a carrier. Okay, this and this fuses, you get XHY, which is what? A diseased male. So here you said that there was hemophilic son. So this is the hemophilic son, and there was a normal daughter. So normal daughter, these both of them, they are normal, but one is what? A carrier. Okay. Now the daughter was married to a normal man. To their surprise, the grandson was also hemophilic. So which daughter was married to a normal man, my dear children? Which daughter would have been married to a normal man? For this grandson to be hemophilic, then it would have been a carrier female. It would have been definitely a carrier female. So this is the daughter, and this is the normal male that you're having. Same thing again, same thing, you're repeating it again. Again you will get a hemophilic son, my dear children. You will get a hemophilic son over here. So represent the cross in the form of a pedigree chart. Give the genotypes of the daughter and her husband. Okay, so you have to represent that form of the pedigree chart and also talk about the genotypes of the daughter and the husband. So daughter's genotype is like this, okay, and that of the husband is like this, normal. Now write the conclusion you draw from the inheritance patterns of this disease. What is the conclusion that you draw? So basically, hemophilia is an X-linked recessive disorder. Okay, that is the conclusion. Males inherit a disease when they receive an X chromosome with defective gene from their mother. Then that's when they get the disease. Okay, females can be carriers if they inherit one defective gene, but they express the disease only if both X chromosome carries the mutation, which is kind of very, very rare. Go through the entire thing. I will be giving you the pedigree. I have not put the pedigree over here. I will be giving you the pedigree later on. Okay, before we start the next chapter. Okay, fine, works. Now going ahead. Identify the genetic disorders given below. So X-linked recessive disorder occurring in, sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of females, leading to defect in the cone of the eye. What is this? This is known as color blindness. This is color blindness. Again, as I told you, whenever X-linked disorder is there, it is the males who are affected because they just have one X chromosome. So whatever effect is there, they will be showing up. There is no one to cover up. You get it? So this is color blindness. 8% of the males and 0.4% of the females are affected. Now sex-linked recessive disorder affecting a single protein involved in clotting of blood. I told you right now it is hemophilia. Queen of England had this disease. Hemophilia. She is a carrier. Okay. Now inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. Accumulation of phenylalanine, that is what you are talking about, phenylketonuria. Now autosome linked recessive blood disease, substitution of amino acid in beta globin chain of hemoglobin. Substitution of amino acid, glutamic to valine. So it is what? Sickle cell anemia. Now autosome linked blood disease, reduced rate of synthesis. Reduced rate, that means quantitative disorder, this quantity. So it is what? Thalassemia. How do you frame the answer? You frame the answer like this. Just go through it. Works? I think that was the last question. Okay, I mentioned it as 26 and 27. I'm so sorry. So that is the last question. The pedigree chart I will be showing you. We have completed this in 1 hour 17 minutes. That this is particular chapter. But you know this chapter is very, very important. Same goes to the next chapter. But the last 2 chapters that we are having, we will make it really fast. Okay, it's just 2 chapters. Okay, works. So I hope I helped you, my dear children. Please take a 5-minute break and come back really fast. I will be here only. You just have to pause the video and go and come back. Just 5 minutes, okay? Drink water, do some exercise and come back. We have to ace the examination. We are going to do principles of inheritance. Again, have molecular basis of inheritance. That is the next chapter. My brain is dead. So let's do that, okay? Take a break and come back really fast. Champions, I hope you're back from your 5 minutes break. So I hope you're fresh, right? let's begin the next chapter, molecular basis of inheritance. Before that, I promised you I will show you the pedigree chart for hemophilia. So here is it how it looks. Okay, so it is an X-linked recessive disorder. So obviously when the males are there, they just have one X chromosome. If that X chromosome is affected, they have higher chances of getting that disease. That is the thing. So females have two X chromosomes. So if one is normal, that normal one will overshadow the effect. So it's the males that is usually affected in more amount. Okay, and as you can see, it's a recessive disorder, which is why it is skipping certain generation. For example, it's not there over here, but in the progeny it's there. So it is sex-linked recessive disorder. This is the pedigree chart for hemophilia. Works, and it's even for color blindness, it would look like something like this. Now depends on who is affected and things like that. Okay. Going ahead, so let's start with molecular basis of inheritance. The first question is, what is the structural unit of a nucleic acid called? Name the chemical components of the unit. So what is the structural unit of a nucleic acid called? It is known as a nucleotide. It is known as what? Nucleotide. Okay, and what does it contain? It contains a pentose sugar. It contains a pentose sugar as in 5-carbon sugar. Okay, apart from that, there is a nitrogenous base, my dear children. Apart from that, there will be a phosphate group. So over here, this pentose sugar is binding with the nitrogenous base on the first carbon. This is second carbon, this is the third carbon, this is the fourth carbon, and the fifth carbon is outside the ring. So the first carbon, the nitrogen base attaches. This link is known as glycosidic bond. This is known as glycosidic bond between what? The nitrogenous base and the pentose sugar. Whereas between the phosphate group and the carbon, what do you call this bond as? Phosphoester bond. What do you call one bond as phosphoester bond? If it is making the similar kind of bond with the next nucleotide, my dear children, which it does. That's how the nucleotides are arranged together. You call this is as phosphoester bond. That also has phosphoester bond. Now two ester bonds are coming, so you call this as phosphodiester bond when there is another nucleotide at the upside. Okay, so each structural unit of the nucleic acid is known as nucleotide. The nucleotide consists of 3 chemical components. The nitrogenous base, my dear children, which again can be categorized into purines and pyrimidines. In purines, you have AG, like AG, OG, you say no, just like that. It is adenine and guanine, this is purines. And pyrimidines, you have cytosine and thymine. Cytosine and thymine in the case of DNA, whereas in the case of RNA, you do not have thymine, instead you have uracil. You have what? Uracil. Please remember that. Now, pentose sugar, it can be deoxyribose in the case of DNA, and it is known as ribose in the case of RNA. You know what? In the case of RNA, over here, there is an OH bond. Over here, there is an OH thingy over here. There is an alcohol group over here. OH group over here, which is why you call this as ribose. Now, you're removing that O, which is why it is known as deoxyribose in the case of DNA. Now, there's a phosphate group as well. I hope that is completed. First question, done. Moving ahead to the next question, mention the 4 nitrogenous bases present in the DNA. I just told you right now, what are the 4 nitrogenous bases? One is adenine, which is a purine, guanine, a purine, cytosine, pyrimidine, okay, thymine also pyrimidine. The other name of thymine is 5-methyl-uracil because it is formed because of the modification of uracil, my dear children. For 5-methyl-uracil is the name for thymine, please not get confused. Now, if you are asked about 4 nitrogenous bases in the case of RNA, you just have to replace the thymine by what? Uracil. Just the uracil. Got it? Cool? Chalo. Next thing. List the nucleosides of the DNA. What do you mean by nucleoside? Basically, I told you there is a sugar, that is a pentose sugar, plus the nitrogenous base. When only them are being spoken about, you call this structure as nucleoside. Now, when the phosphate group comes, you call the whole thing as nucleotide now. So, until then, it is nucleoside. So, deoxyadenosine, that is adenine plus deoxyribose, okay. Deoxyguanosine, as in guanine plus deoxyribose. Deoxycytidine, that is cytosine plus deoxyribose. And deoxythymidine, that is thymine plus deoxyribose. I hope you remember the name of the nucleosides as well, even if the question comes up, and you should be knowing what it means. Okay, you should not go blank. Fine. Next, if a double-stranded DNA has 20% of cytosine, calculate the percentage of adenine. 20% for double-stranded DNA. Okay, that is 20% of what? Cytosine. How much is adenine? So basically, cytosine is something that base pairs with. You know, G and C forms the pair, and A and T forms the pair. Here, triple bond is there between G and C. There are hydrogen bonds, my dear children, and between A and T, there are 2 hydrogen bonds. Cool. Now, over here, if C is 20%, That means the one that it pairs with will also be 20%. Agreed? Will also be 20%. Agreed, no? So, I'll say both of them basically G and C are occupying 20%. And suppose we are taking it as 100. Suppose we are taking the whole thing as 100 and 20% as in talking about. So, in 100%, basically G and C takes 20 and 20%, which is equals to 40%. So, now what about there is one more thing, that is A and T. There is A and T. What about them? So, let's find what is A and T over here. So, A and T, if you want to find out, we'll shift over here. 100 minus 40 is equals to A plus T. 100 minus 40 is what? 60, my dear children. Now, if I say total A and T is 60, then how if you want to divide them? So, I'll say 30% is adenine. Sorry, 30% is adenine, yeah, and rest 30% is what? Thymine. Understood? Over here I have written, so cytosine pairs with guanine. So, if C is equals to 20%, then obviously G is equals to 20%, same. Now, adenine pairs with thymine, the remaining percentage whatever is there will be A plus T. Suppose the total DNA composition is 100%, so I'll say A plus D is equals to 100 minus C plus G, that is 100 minus 20 plus 20 is equals to 60%. Now, if you divide 60 into 2, my dear children, it will be 30. Is same thing I have written over here. It's not be confusing you, but yes, got it? So, it will be 30%. Always remember that, do not do any mistakes. So, what do people usually do is that 20. Now, rest 80 they divide. Don't do like that. So, 20 and 20 is being taken up here itself. That means total is 40. Now, you have to see what is the leftover thing of 40. Okay, that will be 60. Now, 60 also you have to divide into 2. Now, draw a label diagram of a nucleosome. This is a nucleosome, my dear children. In nucleosome, what all do you have? You have a histone octamer. That means histone proteins are there. Okay, and almost 8 of them are there. 1, 2, 3, 4, and then again 1, 2, 3, 4 at the back side. 8 histone proteins are there, and they are very much rich in the positive amino acid, that is arginine and lysine. Please remember the name, arginine and lysine. Why do we need positive amino acids, ma'am? Because the DNA which is there, it is negatively charged, and if something positive is there, probably it might get attracted over there and coil around it. If negative comes together, they will ripple. Okay, but here the DNA is positive and the histone DNA is negative, and the histone proteins are what? Positive, my dear children. So, obviously they will attract, and that's how the DNA coils around it. It coils around it twice, and how many base pairs of DNA would be there? 200 base pairs of DNA, my dear children. And just like after coiling, what happens is that there is another histone protein which comes into play, and that is known as H1 histone. Again, there will be what? 1, 2, 3, 4, 5, 6, 7, 8. Again, the DNA will be coiling around it. The same thing goes on. In between, you have H1 histone. Okay, now what do you call this structure as? Bead on a string structure. As in, on a string, it feels like there are so many beads. Bead on a string structure, hold together, you call this as chromatin. Hold together, you call this as what? Chromatin. When there are strands, you call this chromatin. Now, chromatin can be of two types, euchromatin and heterochromatin. Euchromatin is when they are condensed very much. Okay, heterochromatin is when they are loose. There is space between. So, H1 will have space between two these things. That is nucleosomes. You get it? So, this whole structure that you see over here, it's a nucleosome. Make sure you are drawing the whole thing. Chalo. Identify the following. Pentose sugar present in the DNA. I already wrote it. It is deoxyribose. Okay. Pyrimidine present only in the DNA. The only thing, cytosine is present in both DNA and RNA. The only one that is present is what? Thymine, my dear children. Thymine, yeah, because RNA has uracil. Now, if a DNA 34 nanometer in length has 30 guanine, calculate the number of other nucleotides. Now, this becomes a kind of confusing question for some, so I thought I'll take it up. This is from the question bank, okay. So, 34, so if a DNA which is having 34 nanometer in length, okay, it has 30 guanine. It will be having 30 guanine. You are asked to calculate the number of other nucleotides, that is cytosine, okay, adenine and thymine. How do you calculate it? First of all, we have to see what are the total number of nucleotide bases that are present. We do not know that, right? We do not know that. We just know one thing, that is basically in one turn of DNA, which is almost around 0.34 nanometer in length, this whole thing is one turn, there are 10 base pairs. There are 10 base pairs. That is the only thing that we know. So, we can first find out in this length, how many turns are going to come? So how will we find out? 34 divided by 3.4. We will be knowing the number of turns which are there. So if I divide it, I get 10. So I know there are 10 turns, and in each turn there are what? 10 base pairs, my dear children. So 10 into 10 is equals to what? 100 base pairs are there. I got to understand that there are what? 100 base pairs. Agreed? Now from this 100 I have to divide all this thing. So over here guanine is 30, that means cytosine will also be 30. Agreed? 30 plus 30 is equals to what? 60, my dear children. 60. So we have got 60. Rest of the things will be what? A and T. So 100 base pairs minus 60 is equals to what? 40. Now 40 we have to divide it equally into A and T, right? So this will be 20 and this also will be 20. And the answer is what? 20. Where is the answer? The answer is not written. Somewhere the answer might, yeah, here. Adenine and thymine is what? 2020. Still don't understand? I'll tell you. Please listen to me carefully. Here it is mentioned that if a DNA 34 nanometer in length has 30 guanine, calculate the number of other nucleotides. First thing, we have to see how many number of base pairs are there in total, then only we can find it out, right? So in 34 nanometer length of DNA, I'll say that each turn of the DNA is 0.34 nanometer. So how do I find the number of turns? I divide it. I get 10 turns. Now I know that in each turn 10 base pairs are there, so I calculate 10 into 10 is equals to 100 base pairs. Now I have 100 base pairs. Out of that I'm saying guanine is 30. If guanine is 30, obviously cytosine also has to be 30. So 30 plus 30 is equals to 60. So I'll minus 100 minus 60, I'll get 40. Now 40 I have to divide equally into adenine and thymine, and that's why I get 20 and 20. I will be able to solve this question only if I know this thing. That is, one complete turn of DNA helix is 34 angstrom or 3.4 nanometer. Oh, here I wrote 0.34. I'm so sorry. Okay, I wrote 0.34 here. I'm so sorry. But here I calculated properly. So it will be 3.4 nanometer. Works? Okay? Just give me a second, okay? Yes. So distance between the two adjacent nucleotides is 3.4 angstrom, which is 0.34 nanometer. That means distance between these two base pairs, my dear children, that is what I am talking about, 0.34 nanometer. Okay, distance of full one turn is 3.4 nanometer. Works? Now pentose sugar and phosphate form the backbone of DNA. All this information you should be having in your head, or otherwise you will lose your marks, which I do not want. Okay, so I hope you're enjoying these type of questions, even though this might be giving you a small cute little attack over there somewhere. But please, we have solved it already, so you know how to do the answers, right? So you do not have to worry about anything. So question number 7 is done. Now write the difference between euchromatin and heterochromatin. I just told you right now, euchromatin is when they are like, they have space between them. They are not very much condensed. Heterochromatin is when they are very much condensed. And I'll tell you, this is the DNA which is wrapping around. So you know what, in euchromatin stage, when the DNA is not condensed, at this point of time, something can come and attach. What do you call that something as? What do you call this something as? That is, transcription can happen, so RNA polymerase can come and attach and make an mRNA out of it. So I'll say that euchromatin is something that is transcriptionally active. Transcription can take place simultaneously. But over here, there is no opening. Where will the enzyme come and attach, Baba? So there is no, let's say, transcription happening in the heterochromatin region. So in a typical nucleus, some loosely coiled regions of chromatin with light stain, they take up the stain very lightly, okay, and transcriptionally they are active. It is known as euchromatin. Now the chromatin that is more densely packed and stains dark, and transcriptionally it is inactive, you call it as what? Heterochromatin. You call this as heterochromatin. Now there is one more question. Please remember, it is dark stain, it is light stain, it is transcriptionally active, this is not transcriptionally active. Now name the virus used in Hershey and Chase experiment. Please go through all the experiments. I might not be able to explain all the experiments over here. I might have explained it in the 2 marker, 3 marker and 5 marker, so you can watch those videos as well. As I said, children, there is no option to not watch it. It's very much important. So find out the time for it and watch those videos. You will get your concepts clear. So don't think that if I watch the videos, I am not going to study anything. Arey, you're studying there also. Okay, you're studying there. Not as a teacher, I would not recommend you to watch my video on the day of exam just because of something else. You get it? I know that I have cleared the concepts there, so it will be helpful for you, so you can watch it. And the questions are very, very important. Okay, now Hershey and Chase experiment, in that the virus which was used as bacteriophage, which affects the E. coli. Okay, Going ahead, differentiate monocystronic transcriptional unit from polycystronic transcriptional unit. What do you mean by that? Basically, monocystronic means there is only one gene over here. There is only one gene that will be coding for one protein. That is a monocystron. Polycystron, as in this is a gene over here, this is the area. Over here you have multiple genes, my dear children. You have multiple genes and all the genes will be coding for multiple proteins. So monocystronic contains information for only one gene, whereas polycystronic means it contains information for what? Multiple genes. So there will be promoter, there will be promoter and terminator, and between that there will be so many genes. Okay, whereas over here in monocystronic there will be promoter and terminator but only one gene. Now, monocystronic is found in eukaryotes. We guys have it, okay? But polycystronic gene is found in prokaryotes. The basic example you might be remembering is lac operon. In lac operon, you saw Z, Y, A. 3 of them were coding for 3 different things. It's still there together. So it is polycystronic. It is found in prokaryotes. Lac operon is found in prokaryotes, my dear children. Now, talking about mRNA processing, it requires modification like capping, splicing and polyadenylation. When you talk about monocystronic gene, my dear children, there are so many other processes that you require. You require capping, where you add some chemical groups over here at the 5'N. Okay, you require polyadenylation where you add adenine group at the 3'N. You need splicing to remove all the non-coding regions. So the thing major difference between us and the prokaryotes is that we have certain non-coding regions which are known as what? Introns, whereas they do not have any introns. And the coding regions are known as exons, a very important question. Okay, now the introns before, so DNA is there, DNA is transcribed into something that is known as hnRNA. This is the immature form of RNA. Okay. Now from this hnRNA, capping is done, polyadenylation is done, and the introns are removed. When what do you call this process as? You call this as splicing, removal of introns. And now finally you say that you have got an mRNA which will undergo translation to form proteins. Okay. So here it requires modification like capping, splicing and polyadenylation, but polycystronic in them, they do not undergo any such modification. They do not undergo any such modification which are there, because over here everything happens in the cytoplasm itself. There is no nucleus, there is this and that, but here we have so many things. There is nucleus, a lot of things happens in the nucleus, that is until mRNA formation happens in the nucleus, and then it has to move out to the nucleus. When they move out to the nucleus, the mRNA has to be safe, which is why a lot of things, capping and polyadenylation, everything is done. But here everything happens in the same region. They are like, okay, cool, let whatever happen. Okay, now translation, monocystronic produces one protein, I told you. Polycystronic produces multiple proteins per gene. Got it? That's the answer. Now, write the coding strand and the sequence of bases on mRNA synthesized by DNA template with the sequence 3' Okay, so this is the template strand sequence. You have to find the coding strand, and also you have to find the mRNA that will be formed out of it. So this is the thing, how it looks. So I put the thing over here. So 3' to 5' is mentioned over here. Children, in which direction does transcription take place? From 5' to 3'. So basically from here it will attach and start forming a new thing. So this is the template strand that they have given us. It is in 3' to 5' direction. Please always remember, if they have given ulta, please understand you have to start from 3', reading from 3'. Why? Because RNA polymerase will come attach over here and make a new strand from 5' to 3' direction. Okay. So over here you see this is the coding strand that we have. So entirely complementary strand we have to make. This is a double stranded DNA that you see, my dear children. Over here you have the promoter region, over here you have the terminator region. This is where the RNA polymerase will come and attach and start the process of transcription. Okay. Now, when it transcribes, it forms an mRNA, or basically hnRNA first, as I already told you, but we generally say mRNA. So over here you have to make, A will be going with U this time because it's an RNA. This was a DNA. This strand is a complementary DNA strand, but this is an RNA strand. Remember that. So wherever you had T, you have to replace it with U, and make sure you are mentioning 5' to 3'. I already told you, once more I am telling you, transcription happens in 5' to 3' direction. Now, if the question comes, where is the promoter region located? So promoter is located at the 5' end of the coding strand. You tell like that. Okay. Promoter is situated the 5' end of the coding strand. Terminator is situated at the 3' end of the coding strand. That is how you have to frame your answer. Okay, cool. Works? So this is the answer for this question. Now DNA is more stable than RNA substantiate. How do you express that? So I told you already, in the pentose sugar that it has, okay, in the case of RNA there is a OH group. There it's a OH group which makes it highly unstable. So the second position, this is the first thing, this is second carbon. So RNA has an hydroxyl group at the second position of the ribose sugar, making it more reactive and prone to what? Hydrolysis. Okay, DNA, if you see the pentose sugar of DNA, it's deoxyribose, it does not have this, instead it just has H. O is removed, so deoxy, oxygen is removed, okay. So DNA lacks this group, making it chemically what? More stable, my dear children. Okay, now over here, double-stranded nature of the DNA. So DNA is double-stranded with complementary base pairing, providing structural stability, whereas RNA, it can be single-stranded, it can be double-stranded as well. If it's single-stranded, there are more susceptibility to what? Degradation as well. Now thymine in DNA instead of uracil. So DNA contains thymine, we know that, right? It is more resistant to the mutations. Cytosine deaminates to uracil in DNA. Thymine helps in error correction mechanism. Don't have to write that, just say that DNA has thymine instead of uracil, making it more stable. Okay, so in conclusion, we can say that DNA serves as a good genetic material, whereas RNA, being unstable, is more suitable for what? Small functions like protein synthesis. Not small functions, it's basically short-term functions. Okay, short-term functions like protein synthesis and things like that. Works. Always remember one more thing, RNA, since as I said it has OH group and things like that, it has a catalytic property. It is the one that was used as enzymes as well. So something that can be used as enzyme, I cannot use it for anything else because it has a, it has a chance to go and react with a lot of things. We do not want that. Okay, so these are the reasons that you can write in on the day of your exam as well. I hope it's clear. Yeah. Next. Mention any 2 goals of Human Genome Project. Instead of 2 goals, I have mentioned all the goals so that if it comes in 3 marker also, you can write it. So identify all the genes, obviously, that is first goal in the human genome. So approximately 20,000 to 25,000 genes were considered before, but yeah, it's more than that, right? Now determine the complete sequence of DNA in the human genome. So first is identification of genes. Now that's not all. What are the codes? What is the complete sequence of the DNA of these particular genes, my dear children? So determine the complete sequence of the DNA in the human genome. Third thing, whatever information we have got, we have to store it somewhere. Okay, store information in the database. Okay. Third thing, fourth thing, improve the tools for data analysis. Obviously, after storing it, you should not be like, where did I store it? Oh my God. So you should be having good method for data analysis as well. So you have to improve tools for that. Now transfer related information to other sectors and industry. Obviously you cannot be selfish and keep all the information to yourself. You have to give it to other industry like medical industry and things like that. Okay. Now address ethical, legal and social issues, that is known as LC. Okay. Arising from the genome studies. You have to make sure that you are addressing them. Okay. You cannot randomly kill organisms based on that. So so these are the goals of Human Genome Project, HGP. Works. Next thing, mention 2 salient features of human genome. I have mentioned all the features. Again, this is a question that can come in a 5 marker or a 3 marker, so why we cannot take the risk. Okay, please make sure you are knowing all the things. So human genome contains 3164.7 million base pairs. The average gene consists of 3000 bases, but there is a guy that is dystrophin, it's a protein. It consists of 2.4 million bases. Baba, usually we have 300 bases, 3000 bases, but this is 2.4 million bases. Now the total number of genes is estimated at 30,000, okay, which is much lower than the estimates of 80,000 to 1,40,000 which we thought before. Now the functions are known for over 50% of the discovered gene. Okay, the functions which are there are unknown, are unknown for almost 50% of the genes, and less than 2% of the genome codes for the proteins actually. Rest of them, they are not coding for proteins. Then you might think, no, but they are there. So basically they are there because they are acting as a repetitive sequence. So repetitive sequence make a very large portion of the human genome, my dear children. They are stretches of DNA sequences that are repeated many times, sometimes 100 to thousands. So they are thought to have no direct function, but they have evolutionary significance. Okay, and one more thing, chromosome number 1 has the most number of genes, that is 2968. Chromosome number Y is chutku. Okay, do you know that chromosome Y is chutku? Yes, it has fewest, that is 231. Now, scientists have identified about 1.4 million locations where just single base DNA differences are there. That means between me and you, there's only single base differences in these 1.4 million locations, my dear children, which makes us quite different. Yeah? Yes. So please remember all these points. Now mention the 3 steps involved in transcription in prokaryotes. What is the role of sigma factor and rho factor in transcription of prokaryotes? Okay, 3 steps. You have first thing initiation, where the RNA polymerase will be binding on the promoter sequence, but it says that I want sigma factor, otherwise I would not be initiating things. So sigma factor is very much needed over here, my dear children. So RNA polymerase binds to the promoter region with the help of what? The sigma factor, and starts RNA synthesis. Now elongation is where the RNA polymerase moves along the DNA template, adding more and more complementary RNA nucleotides which will be having uracil, okay, uracil and everything. Termination, at the end when it realizes that everything is over, the RNA polymerase needs the help of some other guy that is known as rho factor. Okay, so it is like crying, I need someone. So it finally comes, rho factor comes and stops this. We don't want this, let's stop this. Okay, so transcription stops when RNA polymerase reaches a termination sequence aided by the rho factor in some cases. Okay, now here the question comes, role of sigma and rho factors, I hope you know that, helps RNA polymerase recognize and bind to the promoter for transcription initiation, that is sigma factor. What about rho factor? It helps, it is involved in rho dependent termination, where it binds to RNA and helps detach it from the DNA template. So these are the points that you need to remember. Going ahead, describe the double helix model of DNA proposed by Watson and Crick. So double helix model of DNA, my dear children, over here, this is the DNA that you have. One strand is in 5' to 3', then the other will be 5' to 3'. It will be anti-parallel, it's not parallel, it's anti-parallel, it's opposite, okay. The 2 anti-parallel polynucleotide chains are coiled in a right-handed manner. They are coiled in what? A right-handed manner. Phosphodiester bonds links nucleotides in the 5' to 3' direction on one strand. So I'll be just showing you a gist of this thing, okay, so that you do not get confused. Suppose this is a this is the sugar that I am having, okay. This is the 5th carbon. On the 5th carbon, you have the phosphate group attachment. This is known as phosphoester bond. Okay. Now, what happens is that there will be another sugar over here, another 5th carbon, another phosphate group. So this one, so this is 1, 2, 3, 4. Similarly, 1, 2, 3, 4, and 5. Okay. Now this phosphate group is also going to attach over here, my dear children, forming what? Phosphodiester bond. This bond together, you call it phosphodiester. This is phosphoester, this is also phosphoester, together you call phosphodiester. So at the end, if there is no one to attach to this P, my dear children, you call this as a free 5' end. Similarly over here, if there is no one to attach to this 3, I will call this 3-OH end. Okay, this is 5-phosphate end and this is 3-OH end that we have if there is no one to attach. So this is 5' and this is 3'. Similarly, you will have it in the opposite way there also. So phosphodiester bond links nucleotides in 5' to 3' direction on one strand and 3 to 5 in the other. Hydrogen bonds between nitrogen bases hold the 2 strands very close. Adenine pairs with thymine, 2 hydrogen bonds. Guanine with cytosine, 3 hydrogen bonds. If at all you are forgetting 2 or 3, always remember G and C looks the same almost. So they have tighter bond. Okay, A and T do not. So it follows Chargaff's rule. What does Chargaff's rule say? That purines, that is A and G, is equals to pyrimidines, that is T and C. Okay. Now, one complete turn of helix is 34 Armstrong, that is 3.4 nanometer, and contains 10 base pairs. Distance between 2 adjacent nucleotides is 3.4 Armstrong, in nanometers 0.34 nanometer. The pentose sugar and the phosphate form the backbone. The sugar and the phosphate which is there, it forms the backbone of the DNA molecule. Fine. Explain Griffith's experiment to explain the transforming principle. What was Griffith's experiment all about, my dear children? So Griffith said, Griffith was in a, okay, there are 3 experiments, always remember. They were trying to find the genetic material. What is the genetic material? Okay, first was Griffith, and then came Avery, MacLeod and McCarthy, and then came Hershey and Chase. So Griffith was the first guy to put on his experiment, and what he did, he had 2 strains of a bacteria, that is Streptococcus pneumoniae. Okay, Streptococcus pneumoniae is a pneumonia. You know it's a bacteria and it causes pneumonia, right? So there were 2 strains of it. One is S strain and the other one is R strain. S strain is known as smooth strain. Okay, why? Because it has a unique polysaccharide capsule made out of it. Okay, they had in the outside layer they had a polysaccharide capsule and it caused diseases. It was the one that caused diseases. Now, S strain is also the virulent form of the strain. It was the one that was causing many diseases, my dear children. It was the one that was not many diseases. Basically, if an organism is affected by the S strain, then for sure it is going to die. Okay. So it is having a polysaccharide capsule, which is why I call it a smooth. Now R strain, if you see, it lacks the capsule and was what? Harmless, my dear children. So it was rough and it was non-virulent as well. Now the experimental setups, what did we do? So we had the little cute mice, okay, and what did we do? We injected the live, let's put this later, I'll put this first. Initially I injected a live R strain, that is the rough strain. And I find that mice was like awful. Hey, what? Nothing has happened to me. I'm just walking around freely, all good. Okay, then they put what? They injected live S strain. That means the one that was live. Okay, that S strain was put into the mouse, and the mouse, like, it died. It what? Died off. And then people were like, wow, how did that happen? So then he thought we should be doing some more experiment. Let's inject heat killed S strain now. As in the S strain, which has been, let's say, put in high temperature and treated with that. After that, you're going to put this S strain, heat killed S strain, into the mice. And the mouse, it survived. It didn't die, it survived. Okay. Now, what did he do? He put heat killed S strain plus live R strain. So until now, live R strain was not doing anything. But when he put heat killed S strain and live R strain, the mouse still died. How? How is that possible? So he was kind of curious to know that. So observation that in the last case, the recovered bacteria from the dead mouse were live S strain. He was very fascinated to know that, and the mouse was died, and he checked what is in this. He found live S strain. But did he put live S strain, my dear children? No, he put heat killed S strain. Then how? And he put live R strain. Then how did he get live S strain at the end? There is something, right? So conclusion was that the R strain which is there, it is getting transformed into a strain due to transforming principle, due to something. It is something that is known as transforming principle. So probably something might have moved out from here and went up here and converted this live R strain to what? S strain. And this something that was moving out was known as transforming principle. This was the genetic material, but they didn't find out. They didn't find out what was it. He didn't find out what was it. He was confused whether it is RNA, DNA, protein, or things like that. So it was later identified as DNA after. Okay, Avery and McLeod-McCarthy, they also did their experiment. Okay, please go check out their experiment. Very, very important. I'm not going to explain over here. I've explained so many times. Okay. So, but I'll just say you the gist. So, DNAs, the enzymes, they were treating with a lot of things. They were treating this thing with RNAs. So, to kill all the RNA. So, if RNA was something that was transforming it, if RNA is killed, if still transformation is happening, then RNA is not, that's something that is transforming it. Then they used protease to kill all the proteins, still transformation was taking place. Then they used DNAs, transformation was not taking place. That means DNA is the one that is transferring from here to here and is the reason for the transformation. Get it? That was done by Avery, McLeod-McCarthy, but that was not approved. Still people had confusion. So, the next guy came in, that is Hershey and Chase, two of them, they came in and they started off with their experiment with bacteriophage and E. coli. Please go through that experiment. Very, very important. Okay. Going ahead. Explain the experiment to prove that DNA replicates semi-conservatively. Children, if you are finding difficulty in finding the experiments, it's there in 2 marker, 3 marker and 5 marker. Please go and watch it. Very important. Okay. Now, explain the experiment. Yeah. So, DNA replicates semi-conservatively. So, how do we explain it? So, suppose I had the DNA, my dear children. This is the DNA. This is the DNA, and this was actually grown in a medium. It was grown in a medium which has N14 isotope. Sorry, N15 isotope. This is the heavier isotope of nitrogen which is there. So obviously nitrogen is a component, okay, of the DNA. So all the nitrogen will be taken up for the formation of DNA. So now the DNA which is there, it has this nitrogen and 15 isotope. Now what did we do? We actually put it inside cesium chloride medium. Okay, we centrifuged it and we found out that DNA was settling towards it down since it is heavy. Since it is heavy, made up of N15 isotope of nitrogen. Okay, this is happening in E. coli. Now what did I do is that I try to grow the same E. coli in a different medium. In a medium that had N14 isotope, which was a lighter isotope. Okay. When I did so, what happened is that basically this DNA strand which is there, obviously this DNA strand which is there, it will divide itself into 2. Here there will be a, oh my god, just give me a second, okay, just give me a second. Okay, I think now it's fine. Yes. So this particular DNA strand which just denatured, it is going to form a new strand over here, but it does have the parental strand over here, my dear children. So the parental strand had what? N15, because both of them were N15, right? But here the new strand which is formed is what? N14. You get it? With that only I understood that it is semi-conservative DNA replication. One parental strand is definitely conserved and a new strand is being formed. But that was not enough, and when I centrifuged again in the cesium chloride medium, I found there is a hybrid layer that is being formed. In the middle it's somewhere forming because it has both the strands, right? Again for 20 minutes again I kept it, total is 40, that is why it is written as 40. Okay, so 20 minutes and then again 20 minutes. E. coli divides after every 20 minutes. I hope you know that. Okay, so over here again I opened up, this guy opened up, this guy also opened up, right? So you get one new strand over here, he also get new strand, he also get new strand. If you see from starting to the end, there will be two strands always that goes from here. That was from here. The parental strand is always being conserved, my dear children. And over here you found a new combination also. N14 was found. N14 was found because when they distributed this guy which is there, again it will be a new strand that will form because we are still growing it in N14 media. So here also you will be finding it, okay, and here in the middle you will be finding something like this, two of them which are like this. You get it? So this was Michelson-Stahl experiment. Please remember the name of the experiment. Was it, what is it? Matthew Michelson and Franklin Stahl's experiment, and they grew E. coli in NH4Cl medium for many generation, where it was N15, okay, that it was used, and then it was N14 that they used, and in cesium chloride they did the centrifugation and everything. Just go through this is important. Fine. Now going ahead, explain the steps involved in translation process. What are the steps involved in translation? What do you mean by translation? So DNA to RNA is known as transcription, RNA to protein is known as translation. What happens in translation is that the mRNA strand will be coming over here. Okay, mRNA strand is over here. Okay, this mRNA strand is having AUG, GGU and all these things there. Okay, now there is someone who has to read this. This mRNA is from 5' to 3' direction, and these things are known as codons. So there should be someone to read the codons. Who are those things, my dear children? This is the tRNA, which is clover leaf in shape, okay, but in actual sense it is like inverted L. It is inverted L. Now if you see the tRNA, it has a 5' end and a 3' end. Always remember, on the 3' end the amino acid will come and attach. What amino acid? Depending on what anticodon they have, or basically, yeah. So basically I say AUG is there, so A goes with U, U goes with A and G goes with C. So UAC, suppose it's there. Over here they will be binding with a pakka amino acid, that is what? Methionine. Always it will be the same. It will be the same. Now this process of binding of amino acid to the 3' end of the tRNA is known as amino acylation. It's known as amino acylation, and that is the first thing that happens. And it is also known as charging of tRNA. It is also known as charging of tRNA. So this thing happened. Now these guys came over here. These guys came over here, right? But can they attach? No. First thing that comes and attaches to the mRNA. So this is happening first, charging of tRNA and everything. Then what happens? The mRNA which is there, it is attached to the smaller subunit of RNA. The smaller subunit of RNA. Okay. Now what happens? This tRNA will come and bind. This tRNA will come and bind, and then comes the larger subunit of ribosome. Not smaller subunit of RNA, I don't know what I am saying. It's ribosome. Okay, ribosome. Here the larger subunit of ribosome will come and attach, and slowly what happens? The tRNA which is there, again a new tRNA will come and bind over here, again a new tRNA will come and bind over here, and thing goes on. Everywhere there is an amino acid. This ribosome is going to move from 5' to 3' direction. Okay, it will move like that. So the amino acids which are there over here, it will break the bond from here and bond with that. So we will get a chain of amino acids as you see over here. So this is the codon that we have. This is the anticodon. The questions can come directly. I have shown you so many questions where they will be asking you what is the anticodon for alanine. For alanine the anticodon is CGU. What is the codon for alanine? It's GCA. You get it? After this, okay, when suppose here, suppose it was, oh, how do I make this? Suppose here it is UAG here. Will translation happen now? No, because UAG is a stop codon. Okay, UGA, UAG and UA. Sorry, I'll write it. UGA, UAG and UA. They are stop codons. Please remember it. Works, and here they are forming the peptide bonds. That's all. So how do you frame the answer? Activation or charging of amino acid. So amino acylation of tRNA is the process of attaching an amino acid to its corresponding tRNA. This is also known as charging of tRNA. Now, initiation. mRNA binds to small ribosomal subunit. The start codon AUG is recognized by initiated tRNA carrying the methionine. Always the start codon AUG, I forgot to say that, AUG is always start codon, and it is basically recognized by the initiated tRNA that will be carrying methionine. The large subunit assembles forming the initiation complex. Now, everything is ready. Now, elongation happens. tRNA brings amino acids to the ribosomes, matching codons on mRNA through its anticodon. Peptide bonds between amino acid, what bond is formed? Peptide bond forms between adjacent amino acid catalyzed by peptidyl transferase. This is the enzyme. Okay. Now, ribosome moves along the mRNA in 5' to 3' direction, I told you. That is elongation. Now, here termination. When a stop codon UAA, UAG, UGA is reached, translation stops as the release factor. What will come and attach over here? Release factor, something else will come and attach over here and stop the freeing the newly synthesized polypeptide which is there. Always remember one very important thing. Ribosome is made up of rRNA, ribosomal RNA, and there are certain ribosomal RNA which has catalytic function of making the peptide bonds and things like that. Always remember that. Okay. So, I have discussed this in 5 marker, 2 marker, 3 marker, so please. Okay, cool. Now, mention the steps involved in DNA fingerprinting technique developed by Alec Jeffrey. So, Alec Jeffrey is the person who basically developed this technique, that is DNA fingerprinting. First thing, you should be remembering that. Second thing, what do you mean by that? So, I told you basically we are same in a lot of sense. Okay, we have lot of common DNA and things like that, but there is one thing that is different in between us. We have different types of repeats. So, in our when we studied about the salient features of the genome, you found that there are so many repetitive sequences in our body. So, in my body, for example, it is AGCT. In your body, it can be TAGGC. You get it? So, we have unique set of repetitive sequence, and they are repeated in very much amount. Now, I want you to think of a scenario. There is a murder that happened. Okay, there's a murder that happened that I did. Okay, Jasmine ma'am did. Okay, now that's nothing to glorify it. I am going to get caught. I am going to show you how. So, basically, I did a murder, and now my blood is there. Okay, now the police came and they found the blood, and they were like, we'll check this up. We are going to check this up. So, they checked this up, and they found out, okay, this is the DNA which is there in the cells of those cells, or the blood which is there. So, the DNA was found out. Okay, now what did they do is they made a complementary strand out of it. So, this was the DNA, they made a complementary strand out of it. I will call this as a probe now, because I am going to attach it with something that will be, let's say, radiating, it's emitting its light or things like that under UV radiation and things like that. Okay, so this is the complementary strand that was formed. It will definitely have my repetitive sequence, which is very unique to me. I am unaware of all this thing. Now, me and my friend Chintu, okay, we both are here. We are the culprits. Now, Chintu didn't do anything, but I am kind of framing Chintu so that he did the murder. Police was like, let's do a DNA checkup. So, they took my DNA and Chintu's DNA. Okay, they took my DNA and Chintu's DNA, and they basically isolated the DNA first thing. Obviously, you have to isolate the DNA because there are proteins and everything up there. So, isolate it, and then you're going to cut it with restriction enzyme. Okay, you're going to cut it with same restriction enzyme which was used up over here. Probably you can use the same restriction enzyme for hits also, and you're going to cut the DNA strands. Got it? You're going to cut it, and then after cutting the DNA strands, you're going to separate them. You're going to separate them on a gel electrophoresis chamber. So, this is a chamber, this is mine, and this is Chintu's. Okay. Now, what was done? Basically, whatever is there over here in the gel electrophoresis chamber will be transferred on a nitrocellulose sheath, and this is known as southern blotting. I take everything on the southern blot. I have the southern blot with me here. Now, I take my probe which is there and make it run through it. What happens is that probe is a complementary thing. So the DNA which is there, if at all it is there over here also, the probe will go and attach there. So when found, it was found that the DNA is finally going and attaching over there, my dear children, on the probe. That means what? I am the culprit. It does not go and attach to Chintu's, because he is not the culprit. You visualizing it how? You are going to visualize this fluorescent drops by basically using an X-ray film or a UV light. Similarly, the same thing can be done where the DNA can be just cut, made through electrophoresis chamber, and then we are just going to compare. Suppose I have over here, when will I compare? I don't have space, my dear children. Okay, so there is a new technique that I learned, but it will be writing everything that I say. So anyways, suppose, oh, it does not work here, does it? No. Okay, let's drop it. So basically, anyways, so I am removing Chintu's, okay, I am removing Chintu's for the time being. Suppose this is the exact band that I got over here. That means I am the culprit anyways. So both of this thing can be used for DNA fingerprinting, my dear children, which is very unique to us. First thing, isolation of DNA, and finally I am caught, okay, I am caught, so please not do any murders. So isolation of DNA, DNA is extracted from the sample, that is blood, hair, saliva, etc. Okay, please remember these things, blood, hair and saliva. Now, digestion by restriction enzymes. So DNA is cut into fragments using specific restriction enzymes, and then separation by gel electrophoresis. The DNA fragments are separated based on size using agarose gel electrophoresis. Then transfer to membrane, that is known as southern blotting. Separated fragments are transferred onto what? Nylon or nitrocellulose membrane, and then you hybridize with DNA probes. Radioactive or fluorescent probes complementary to the specific sequences bind to variable regions of the DNA. Visualization. The hybridized fragments are visualized using X-rays or UV light, producing what? A unique DNA pattern. And this pattern is very unique to us. Okay, I hope you got it. This is DNA fingerprinting technique by Alec Jeffrey. Now, the following sequence of nucleotides, this in mRNA codes for a sequence of 6 amino acids, generate the salient features of the genetic code. First things first, from where do you start reading the genetic code? You tell me. From here? Ma'am, this is a start code and we'll start from here. It started from 5' to 3'. So it will be going from here to here, my dear children. It will be going from this direction to this direction, right? So 5AAU, then GAG, then GGA, then UUU, then CAU and GUA. That is the thing. That is how you read it. What is so unique about it? First thing, if you see, all the codons, my dear children, are in triplet format. Are in triplet. Okay, there are 64 codons in total. 61 codons code for the amino acids. 3 codons, there are stop codons, they do not code for anything. Now, even if it's not mentioned over here, let me just tell you. Okay, let's see what is here, then we'll come on to what is not there. So some amino acids are coded by, that is also not there. The codon is read in RNA in a contiguous fashion. Yes, the codon which is there, it is read in a continuous fashion. That can be said from here. So that is one point. Now UAA, UAG, UG are terminator codons, that is also not been mentioned over here, so I won't say. Now what else is there? Yeah, there is one feature which I have not mentioned over here, and that is unambiguous. The codons are unambiguous. That means one codon which is there, it will be coding for one particular amino acid only, nothing else. So from this I can understand the codon is triplet. Yes, I can understand that. And then the codon is read in mRNA in the contiguous fashion because there are no punctuations, and it is unambiguous because one codon, as you see over here, for example AAU will be over here. So AAU, my dear children, it will always call for asparagine, no, nothing else. Okay, please remember that, asparagine and nothing else, right? So that is one thing. So yeah, so these 3 things are there, and the codon is nearly universal. For example, from bacteria to human, you would code for phenylalanine, except there are some exceptions, that is mitochondrial codons and also some protozoans. Now the features which are not mentioned over here, let's talk about that. So there is AUG codon, it has dual function. One of it is to code for methionine, and the second function is to basically act like an initiator codon. From there only initiation of the process of transcription happens. Now there are some amino acids which are coded by more than one codon. Remember, this is not opposite of unambiguous. Please do not think like that, okay? This is not the opposite of unambiguous. Degenerate means one particular codon So multiple codons will be there for one particular amino acid. Multiple codons will be there for one particular amino acid. This is known as degenerate. Okay, all the codons are coding for one amino acid only, that is for sure. I am telling you there are multiple codons for one amino acid. You won't see CUU coding for anything else apart from leucine. So that feature says that it is unambiguous. It is not going to code for any other amino acid. But I am also saying that leucine is coded by multiple codons. That is known as degeneracy. Okay. Now, yeah, rest all things I think we have covered. UAA, UAG, UGA are stop terminal codons. That's all. So, in context to lac operon, when Jacob and Manott cultured E. coli in a medium devoid of lactose, E. coli cells formed colonies, and when lactose was added, E. coli cells metabolized lactose by synthesizing enzymes. So, here is the story of lac operon. It's a very fun story. Children, if you remember, I told you that in the case of prokaryotes, they have polycistronic transcription unit. As that means you have multiple genes in one particular cistron, right? So, you have ZYA gene over here, and there is a promoter region for that particular gene. So, the RNA polymerase will be coming and binding over here, and further will be helping in the transcription of all these genes. Now, let me tell you one very important thing, that is there is an operator region as well over here. What does this operator region do? I am going to talk about that, but before that you should know what is this. So, this is also a gene. This is known as a regulatory gene. What do you call it? Regulatory gene. It has its own promoter sequence, my dear children. Okay. Now, this regulatory gene can regulate positively and negatively, but here this guy is regulating negatively. It is going to produce a repressor protein. What protein? Repressor protein. And this repressor protein will go, run and bind on the operator region. Now, you only think, if something comes and binds on the operator region, the RNA polymerase which is going to come and bind on the promoter region, will it be able to move forward? No, it won't be able to move forward. So, does this transcription happen? No, this transcription would not be happening. And transcription of what? What are these genes coding for? So, first is Z gene. Z gene is coding for beta galactosidase. You know what? Whenever E. coli feels that I do not have energy, so basically if it is put in lactose medium, it tries to take this lactose and break it down to glucose and galactose. That is how it gets its energy. Now, who helps in taking inside the lactose? It is permease enzyme. Permease enzyme basically increases the permeability of the membranes of E. coli and let the lactose inside. Now, during this entire process, there will be certain byproducts being made. Who are the ones who are going to tackle with the byproducts? It is the transacetylase enzyme which is there. So, all these three enzymes are really important, but over here, since the repressor is binding on the operator region, the RNA polymerase is not able to go ahead and transcribe all these things. Now, that is a scenario. So, I'll say as of now, the lac operon is in switched off condition. What? Switched off condition. Now, if I want to turn it on, what do I need? Basically, I let the lactose inside. Now, when the lactose comes inside, my dear children, it is going to bind on the repressor protein. So, it is acting like an inducer. Lactose or a different form of lactose which is known as allolactose, both of them, either of them, okay, can act as an inducer and can bind with the repressor protein. Now, when it binds with the repressor protein falls in love with it and forgets everything, that I have to do my work, that is of going to the operator and stopping the transcription. So, this guy is now free. Transcription happens and beta galactosidase, permease, transacetylase, everything is made. Finally, the lactose which is there, it is also broken down, and that's how energy is received. So, this is switched on condition. This is what? Switched on condition of lac operon, where the operator region is not bound by anything. Now, you might be having this doubt that where did lactose come in? For letting lactose inside, you need a little amount of permease, right? So, yes, you know what, when we say that it is stopping the process of transcription, it's not like entirely stopping, even though it's not written in the NCERT, I'm telling you, it's not entirely stopping, little amount of transcription still happens. So, permease is still being produced, so that's why lactose or allolactose gets inside and it turns on the lac operon. I hope you got it now. Now, over here, so, this is the switch off mechanism and this is the switch on mechanism. Switch off as in when lactose molecules are absent in the nutrient medium, DNA is switched off for protein synthesis. Okay, nothing is happening. Now, the regulator gene produces a kind of protein called repressor that binds to the operator gene. This do not allow RNA polymerase enzyme to transcribe the structural genes. Yes, structural genes as in ZYA. Now, hence, there is no transcription, translation and required protein is not synthesized. Please remember, when I am saying all this thing, some people they forget what does ZYA Z stand for, Y stand for, A stand for. So all the silly things that is there, my dear children, small things is very important. Please do not lose your marks there. So Zia, you can remember like Zyada. It's a Hindi word, to more, okay. So Zia, you can remember like that, Z-Y-A. So it is first is beta-galactosidase, then permease, and then transacetylase. Now hence it is called switched off mechanism. Now switched on, the lactose molecules added in the nutrient medium acts as an inducer which binds to what? The repressor, to form inducer-repressor complex. Now the repressor becomes inactive and released from the operator gene. This enables the RNA polymerase enzyme to transcribe all the 3 structural genes, that is Z, Y, and A. Now all these 3 mRNA strands undergo translation to produce 3 polypeptide chains or 3 proteins which acts as 3 enzymes such as beta-galactosidase, permease, and transacetylase. These 3 enzymes together break the lactose into what? Glucose and galactose, and finally the E. coli gets the energy. Got it? Chalo. Next question. Name the process involved in the following. Okay, first process says one parental DNA strand is conserved in daughter cell. What is it? Semi-conservative mode of replication. Now, separation of DNA fragments. So, separation of DNA fragments in the gel is known as gel electrophoresis. It is known as gel electrophoresis. Now, separation as in DNA fragments separating from a, let's say, double helical structure to single stranded, that is known as denaturation. But over here, I suppose they are talking about gel electrophoresis. Okay. Now, removal of introns in H and RNA, heterogeneous RNA. What do you call this process as? Removal of introns. You are removing the introns. You call this process as splicing. I told you, introns are something that do not code for anything. We do not need them. So, it's okay to remove them, and this process is known as splicing. Now, attachment of Rho factor to RNA polymerase during transcription. Rho factor is needed when it's finally tired. RNA polymerase want to end the transcription, right? So, it is for termination of transcription. Right? Now, polymerization of amino acids to form a polypeptide on mRNA. What is it? Translation, polymerization of amino acids, as in you're bringing the amino acids together, and this is known as what? Translation. So, here are all the answers, my dear children. You can just go through. It will be really helpful for you. Cool? Now, with this, I remember one more thing. Please remember the type of RNA polymerases in human, in what say, eukaryotes. You have polymerase 1, you have polymerase 2, you have polymerase 3. What are they going to make? Very important thing. So, in the transcription part only, you can see it. Please go through it once, or you can refer the video to 3-marker, 5-marker video. In that also I have explained it. Yeah, I think so. Yes. Now, a policeman from the crime department approached the forensic science department to help identify the culprit. Me, remember Chintu and me? Yeah. The DNA samples were collected from the crime site, and of the 2 suspects, explain the process involved. DNA fingerprinting is the process that is involved. Now, if the culprit had monozygotic twin, can this method give results? So, Jasmine and someone else, okay? I had a twin, suppose. Do you think this method, which is DNA fingerprinting, can it give results? What do you think? No. You cannot. Monozygotic, as in basically there is one zygote which is formed on the fusion of egg and the sperm. Okay, you form the zygote. Now this zygote is dividing, separating into 2 and forming 2 different organisms. So every genetic material which is there, my dear children, it will be same in both of us. All of it will be same in both of us. So obviously there are no chances that we can find with using DNA fingerprinting. So monozygotic or identical twins have the same DNA sequence, making the standard DNA fingerprinting ineffective in distinguishing them. Please remember that. Okay, cool. Okay. If it was dizygotic, then dizygote as in 1 egg, 1 sperm, 1 egg, 1 sperm. Over here it is forming a zygote, here it's forming another zygote. There is a difference, because in all the gametes there can be some amount of differences there. So yes, even though it's produced from the same parents, there can be some differences. Fine, but monozygotic twins, no, big no. Cool? Chalo. Now, which sequence of bases, okay, for here, explain the process involved, we have already explained DNA fingerprinting, please refer to that only. Now, which sequences of bases transcribed from DNA are found both in hnRNA and mRNA? These are the questions that are added up at the last moment as of now, because I wanted you to have the memory of this, that you have to remember it, okay. So, the sequence of bases transcribed from the DNA that are found both in the hnRNA and mRNA, as in this is the DNA. From this, when I transcribe, I get hnRNA. This hnRNA is going to get processed and to form and then it forms the mRNA. So what is that? Something which is found in both hnRNA and mRNA, that is the question. So it will be what? I told you, all the exons and the introns are going to stay up over here. Everything, we will be having everything over here, exons and the introns, everything we would be having over here. Now in the case of mRNA, we are going to remove these introns. We are going to remove these introns. We just have the exons which are joined up. Okay, so the sequence of bases transcribed from DNA that are found both in hnRNA and mRNA are exons. Exons are coding regions that remain in the mRNA even after the process of splicing. The removal of introns is known as what? Splicing. Okay, now while introns are removed during processing. Works? This is the thing, introns are removed and then you get mRNA. What is common? You have exons. Now, mention the levels where gene expression can be regulated in eukaryotes. I was telling you, right? I was telling you in what all levels can it be regulated. So, in transcription level, my dear children, that means formation of primary transcript. At that point, you can stop the process of transcription and regulate. Second thing, processing level, as in I told you, if you have to form an mRNA, you have to do splicing and all. So I can regulate in that level also. I do not allow the processing to happen. Then what will happen? I can regulate it. I won't allow the splicing to happen, then it won't go ahead for translation, right? So that is one thing. Now transport of mRNA, that is another thing. It was all happening in the nucleus. Now finally, if it is moving out, the transport of mRNA from the nucleus to cytoplasm, that is also one thing. Then you have translational level. You have what? Translational level, where the mRNA which is there, suppose it do not undergo the process of translation. Okay, the tRNA does not come and bind, we stop it somewhere like that, then again translation can also not happen. So these are different levels in which you can regulate gene expression in the case of eukaryotes. Yes? Now, mention the criteria for a molecule that acts as a genetic material. What are the criteria for the molecule to act as a genetic material, my dear children? It should be able to replicate. For example, it won't say that I am going to have just one copy, I am not going to divide to anything. Is it like that? No, obviously it has to make multiple copies. It has to pass down in every cell. So it should be able to generate its replica. Second thing, it should be chemically and structurally stable. For example, I am a person who go and becomes friend with everyone. In that case, what will happen? Will I have a permanent good friendship? It's like, no, that's not a good example. I can make multiple friends. Simply, just let's take it simply, okay? Let's not give example. So over here, if I am not chemically and structurally stable, okay, for example, I do multiple jobs. Here also I teach, there also I teach, I do everything. Will I be able to concentrate over one thing? Can I be stable over here? No. When you need me, will you get me? No. So that is the thing. So it should be chemically and structurally stable, my dear children. Otherwise, it's a problem. Yeah, now it should provide the scope for slow changes. Obviously, it cannot stay like that. I won't make any changes. I cannot be so adamant like that. There should be some changes in me, very slow changes in me that would make myself better. So there should be what scope for slow changes that is required for what evolution, and evolution is always mostly for better things. Now it should be able to express itself in the form of Mendelian characters. Okay, it should be able to express itself in the form of Mendelian characters that Mendel put forward. That is dominant, recessive, and all these things should be maintained up there. Next, will you remember this? These are the points. Please not forget this. Please. Chalo. And finally, we are up to the next chapter, that is ecosystem. Ecosystem does not have many questions up there. Okay. So are we ready? You're ready. Chalo. First question, list the 4 primary functions of an ecosystem. What is the primary function of an ecosystem? First is to produce food, right? First is to produce what? Organic matter, my dear children. What do you call this process as? You call this as productivity. So ecosystem produce biomass through photosynthesis, right, in plants, and then consumption by organism, that is secondary productivity. So for example, the plant that produces food and everything, this is known as primary productivity. Now suppose I am going to eat the plant. Okay, and now whatever food I have in my body, total thing is what? The secondary productivity. Some other organism, for example, tiger comes and eat me, it gets this. So productivity is really important. That is one major function of what? Ecosystem. Second thing, decomposition. Breakdown of organic matter by decomposers into simpler inorganic matter. That is also one very important primary function of what? Ecosystem. Third thing, energy flow. You need energy flow to happen, right? So that is also a function of ecosystem. Transfer of energy from the producers to the consumers, primary, secondary, tertiary, all the things. Now, nutrient cycling, recycling of essential nutrients, or the nutrients, if they say that I am going to sit over here, not moving anywhere, will you be able to survive? No. So recycling of essential nutrients like carbon, nitrogen, phosphorus between biotic and abiotic components are really important. Okay, so these are the 4 primary functions of an ecosystem. Since the answer seems like it's of something else, okay, you might find it difficult to remember, which is why I'm telling you, remember it, try to remember it as such. Functions of ecosystem, as in what does it do? Productivity, it helps in productivity, it helps in decomposition, otherwise things will be like this only. It helps in energy flow, it helps in what? Nutrient cycling. Thanks. Okay, next, define gross primary productivity and net primary productivity. What do you mean by gross primary productivity? For example, I'm a plant. By photosynthesis, I am producing some organic matter. By photosynthesis, I am producing some organic matter. This is gross primary productivity. Now, since I am a plant, I have my own needs. I have to basically make some apples, okay, and basically, I have to respire, I have to do a lot of things. So, I'll say that I am going to use some of it for my own things. So, I'll call this as respiratory losses. I'll call this as respiratory losses. So, gross primary productivity minus the respiratory losses is something that I have left to give for the next organism that is going to eat me, and that is known as net primary productivity. That is known as net primary productivity. So, how do you frame the answer? Gross primary productivity is the total amount of organic matter or biomass produced by what? The producers through photosynthesis in a given area and a time. In a given area and a time, whatever amount of organic matter is produced by the producers through photosynthesis is known as gross primary productivity. Now, net primary productivity is what? The remaining biomass after subtracting the energy used by the plants for respiration. That's what I told you. NPP is equals to GPP minus respiration losses. Respiration losses means everything. For whatever I'm doing, for example, for my metabolism, if I'm a plant, I'm using this energy for my metabolism, for for exchanging of gases, for reproduction, whatever. After that, whatever is left behind is known as net primary productivity. Now, this is the energy available to what? Herbivores and decomposers. Please remember that. Okay, so this is NPP and that is GPP. Next question. Mention 2 conditions which increase the rate of decomposition in ecosystem. With this question, I want to tell you the steps involved in decomposition is really important. Please remember, first one is fragmentation. First one is fragmentation, where for example, I am a earthworm. I'm going to eat everything up and then I'm going to poop out things. Okay, so basically this earthworm is breaking down big things into smaller particles, and this is known as fragmentation. This is a physical process. Okay, now after fragmentation, what happens? Basically, there is something that is known as leaching. So, whatever things are there which can be moving down to the soil, it will be moving down, and that is known as leaching. It is not going to be available for any process. It's just there. It is just going to seep down. It's just going to store there. Okay, that is known as leaching. Now, there is something that is known as catabolism, where finally, this big things broken down to small things, whatever is left behind, the organic matter which is there over here is going to get broken down to what? Inorganic matter. Is this a one-step process? No. There are so many steps involved over here. Now comes humification, where, as I told you, it's not a standard process, right? So, when organic matter breaks down into inorganic matter, there comes a middle, time where it is forming something. It is going to form something that is known as humus. That is known as humus. And humus is very much rich in like mineral nutrients and everything. Slowly, this humus finally will be breaking down into complete inorganic nutrients, my dear children, and this is known as mineralization. It is known as mineralization. So, I can say that humification and mineralization comes under this, but I would not. I would rather put this in this order. For fragmentation, first, leaching second, third, catabolism, fourth, humification, fifth, mineralization. Now, over here, you're asked, what are those conditions which increases the rate of decomposition? What are those conditions? First is warm temperature. High temperature enhances what? Microbial activity, speeding up the process of decomposition. That is one step. Second thing, high moisture and oxygen availability. So, adequate moisture and oxygen promote the growth of decomposers like bacteria and fungi, accelerating the breakdown of organic matter. Very important point. There should be high moisture and there should be oxygen availability because there are some aerobic bacteria who has to break down things. So, adequate moisture and oxygen promote the growth of decomposers like bacteria and fungi, accelerating the breakdown of what? Organic matter. So, warm temperature, high moisture and oxygen availability. Very important, can be asked in the form of MCQs. Okay. Next, why is the pyramid of biomass in a sea generally inverted? Biomass, pyramid of biomass in a sea, why is it generally inverted? To tell me about pyramid of okay, here I'll tell. Okay, this is another question, but still. So pyramid of biomass, biomass means whatever is left, okay, the whole everything. So over here, in the case of sea, who occupies the lowermost trophic level? It is phytoplankton. And they are very chutku organisms. Now phytoplankton. अच्छा ठीक है मैंने pause. Okay, wait. So sorry children, so there is some celebration happening up over here, which is why that pause happened. So yes, so there is phytoplankton in the case of oceans, okay, or sea, where they are the primary producers. Now phytoplanktons are very chutku people. Now they are eaten up by something else that is known as zooplankton. Okay, but please understand the biomass of each phytoplankton is really less. When you talk about zooplankton, it will be obviously way more than the one that is there for phytoplanktons. Okay, now going ahead, zooplanktons are eaten by small fishes and small fishes are eaten by large fishes. Now you tell me, don't you think the biomass is way too much for them and way too less for this? Yes, which is why there is an inverted biomass pyramid that you see in the sea. Okay, that you see in the sea. Okay, so what is written over here? Why is the pyramid of biomass in sea generally inverted? Short lifespan of phytoplankton. Phytoplankton which are there, they have very short lifespan also. So obviously if you count the biomass, you have to see what are all living and things like that. So basically they have a very short lifespan, which is why they are going to die and their new ones are going to form. So if you see the overall biomass calculation, it becomes accounts to be really less. Now higher biomass of consumers, zooplankton and higher trophic levels accumulate more biomass over time compared to what? The poor phytoplanktons which are there. Now rapid consumption, phytoplanktons are consumed really fast. Again, if you see, if something is consumed really fast, do you find it there? No. Suppose I have 5 biscuits. Of all the 5 biscuits, if I consume it, will I have any biscuit over there? No. Just like that, zooplanktons are going to consume the phytoplanktons really way too much, which is why they will be having less biomass. Cool. Now the next point, pyramid of energy is always upright and can never be inverted. What do you mean by upright and never be inverted? So my dear children, please understand this thing, that for suppose this is the trophic level 1 where you have plants. Now plants are eaten up by rabbits. So rabbits occupy trophic level 2. Now rabbits are eaten up by tiger or probably human beings. Okay, let's see human beings. So it is trophic level 3 and the human beings are eaten up by tiger. That is trophic level 4. Now trophic level 1, I say it always has the highest amount of energy, that is for example 10,000 kilojoules of energy. How much amount of energy will be transferred to the rabbit? Only 10% of 10,000, my dear children, because as I said, the plants will be utilizing something for themselves. Whatever is left back could be given to the next trophic level, and there will be certain losses also. There will be certain respiratory losses also. Okay, so 10% of 10,000 is what? 1,000 is what? 1,000 kilojoules. Now again, 10% of 1,000 will be what? 100 kilojoules. Now 10% of 100 will be what? 10 kilojoules. So if you see, if you go above the trophic level, it is obviously gonna decrease. So which is why it cannot be inverted. It cannot be inverted. And there is one very big reason for why this inversion is not possible. The sun's energy, can we trap it? Can we trap the sun's energy? No, my dear children. There is only plants who can do that, which is why they will be having the highest amount of energy. Now, where you have the highest amount of energy, those organisms can only pass it down to the next organisms which are there. Got it? Did you understand it? So, yes, please go through this particular answer once more. So over here, energy flow is unidirectional. Please just go through this, okay? This is very important. Yeah, so the first point is energy flow is always unidirectional. Energy is transferred from producers to consumers, but portion is lost as heat at each trophic level. One thing. Second thing is loss of energy at each trophic level. That also happens because all the organisms that you see, my dear children, they have to utilize some for themselves. Then only they can give for rest. So if I'm utilizing, for example, I had got 4 biscuits, I had 1 biscuit. Now you would get what? 3 biscuits. So obviously I'm utilizing something for myself. So obviously it is going to get lesser. Okay. Now producers have maximum energy. Why? Since they capture solar energy, my dear children. So the base of the pyramid always have the highest energy. Now no energy transfer occurs backwards. Unlike biomass, energy cannot be recycled back to the lower trophic level. What is lost is lost. Please understand that. So these are the reasons why energy pyramid is always upright and not inverted. This is a very important question. Okay. Going ahead with the next question that we have, that is schematically represent pyramid of biomass with appropriate units. So let's say the pyramid of biomass for C, it is inverted for sure, it looks like this for sure, but the normal grassland areas in that, or basically the forest and all, if you talk about the forest, okay, so there are so many trees and trees have lots and lots of biomass, my dear children. So I will say over here the biomass will be way too much. Here the producers have way too much biomass, then comes primary consumer, secondary consumer and tertiary consumer. So this is the type of biomass pyramid that you have to make. Make sure you're making like that itself. Yes, I'm so sorry, there are so many disturbances coming along. I don't know why and where have they come from, but yes. Now, define the terms food chain, food web, trophic level, stratification and standing crop. Food chain means you know. So there is a chain of a linear sequence of arrangement of what organisms, my dear children. For example, there is plant, plants are eaten by rabbit, rabbits are eaten by humans, humans are eaten by tiger. So this is a linear sequence of organisms which we have. We do not have branches in food chain. We do not have branches in food chain. Okay, but when you talk about food web, we have branches. For example, plant being eaten up by rabbit, rabbit can be eaten up by human beings, and rabbit can also be eaten up by a tiger. So you see a branch there, and this tiger can be eaten up by a lot of things. Okay, basically the dead tiger probably. Yes, so get it, this is how things work. So this is known as a food web, where you have interconnecting food chain. So food chain, when you just talk about food chain, it's just a linear sequence of organism. When you talk about food web, it's like a connection, interconnection of what, so many food chains together. Okay, now what do you mean by trophic level? Trophic level is a specific position that an organism occupies in a food chain. For example, I say you have producers, that is the plants, and they are eaten up by, what do you call, they are eaten up by rabbit. So the position that this plant is occupying in the ecosystem, okay, it is known as trophic level. This is known as trophic level. In this food chain, what is the position? That is a trophic level. Fine. So the specific position an organism occupies in a food chain based on its source of energy, such as producers, primary consumers, etc., are trophic levels. Now what do you mean by stratification? It is a vertical distribution of different species. So you might be seeing that certain plants or certain organisms do not need too much light. Okay, so they might be in shaded areas. Some, they need light, so they might be in taller areas. So vertical distribution of different species in an ecosystem based on light availability, temperature and resources is known as stratification. Okay, so ground layer, then understory, then canopy, all these things come under different layers. Now standing crop, the total biomass of living organisms present in a particular trophic level at a given time. So total biomass of living organism present in what? A trophic level at a given time. What is the biomass of the total number of producers which are there, okay, at a given time? That would be the standing crop for that. So total biomass of living organism present in a particular trophic level at a given time. That is the answer for this. Got it? Now, ecological pyramids have limitations. So as of now, we spoke about pyramids like pyramid of energy and this and that. So all of them, if you saw, did you see anywhere that I spoke about decomposers? No. But do decomposers play an important role? Yes, because decomposers are the one that recycle the nutrients. If decomposers are not there, there is no one to recycle the nutrients. If nutrients are not there, do you think plants can grow? No. So whatever dead and decaying things are there, whatever nutrients are stored in them, it has to be put down into soil. Who does it? Decomposers. So if decomposers are not there, are we providing the nutrients to the soil? No. Can we build a new organism from the soil, that is plant? No. So role of decomposers has not been mentioned. That is one very big limitation of ecological pyramids. Then they do not account for seasonal variation. That is, biomass and energy flow can fluctuate with seasons, right? It can fluctuate with season. In different seasons, it can grow too much. In different seasons, it won't be growing too much. There could be fluctuation in that. They are not worried about that. They are not mentioning that as well. This you can put as a third point. Second point you can put as this. Same species at multiple trophic levels. Same species at multiple trophic levels. For example, I have an omnivore organism. So either it can directly feed a plant or it can feed on organism that feeds a plant. Didn't get me? So listen, there is plant and there is X organism. This X organism can feed on the plant, or probably this X organism can feed on Y organism that feeds on plant, because this X is an omnivore. It can eat both the plants as well as other organisms. You get it? So you're not talking about that also. Some organisms feed at different trophic levels, making strict categorization is actually difficult. You cannot categorize them that they will be always primary consumers. You cannot do that. Now fourth thing, do not show energy transfer efficiency. Pyramids represent trophic levels but do not indicate the exact efficiency of energy transfer between the levels. This can be the last point that is there, okay? But make sure this is the second point and this is the first point. Works? Okay? And with this, I think we have completed the chapter, that is ecosystem. More questions would be found, as I said, it will be found in 2 marker and 3 marker. There are no 5 marker questions coming from this chapter because the weightage is 4 marks, just 4 marks, which is why I am also not going ahead deeply into the concepts. Okay, but I hope you got a basic understanding of the questions. Okay, any other question that could be coming from it is, I think that's all. The decomposition process I told you, please go through it. It's very important. Okay, now we'll go to the next thing, that is biodiversity and conservation. Very important chapter. Okay, very important chapter. As a scoring chapter. Why to lose your marks? Because the questions are way too direct, so there is no chance to lose your marks. Please, I'll recommend you for this particular chapter, read your NCERT properly. Read your NCERT. I wrote NEERT, please don't write NCERT, okay? Read it properly. There are certain technical terms, that is 8.1% of this and that percent of that, that could be important. So just go through it once, okay? Now, over here, what is genetic diversity? India has more than 50,000 different strains of rice. Mention the level of biodiversity it represents. So this is a question just about genetic diversity, but I have mentioned all different kinds of diversity because if the question comes, what are the different type of diversity, you should be knowing it. So genetic diversity, which is being asked over here, it means variation within a species at genetic level. For example, I am talking about rice. You have so many different types of rice. Aren't you talking about the same species? Yes, that is rice species. You have basmati rice, you have this rice, you have that rice. It's the same species of rice only. You're talking about rice, but within a species, right? You're talking about rice which is the same kind of, oh my god, wait. Jimbomba, let's forget everything. Listen to me carefully. So, I'm not talking about different type of rice and wheat. I'm talking about different types of rice. Within the rices, you have different varieties. I'm talking about that. So the variation which occurs within a species itself at genetic level, that is known as genetic diversity. Variation occurring within a species at genetic level, that is known as genetic diversity. Example is raw Wolffia vomitoria, shows genetic variations in racemine content. In the racemine content throughout, okay? Now, apart from that, India has 50,000 rice strains, which is being mentioned over here, and 1,000 mango varieties. 1,000 mango varieties, my dear children. All of this is an example of genetic diversity. Now, going ahead, you have species diversity. So, diversity at species level. Example, Western Ghats have higher amphibian diversity than the Eastern Ghats. Diversity at the species level, my dear children. So, you're talking about 2 different locations and you're saying that basically Western Ghats have more amphibian diversity than the Eastern Ghats. To 2 locations, same type of organism, but the diversity is way too different. Okay, so diversity at species level is known as species diversity. Now, ecological diversity, for example, I say in India we have tropical, we have temperate regions, we have desert, we have tundra, a lot of things. So, diversity at ecosystem levels, we have different type of ecosystem, wetland ecosystem, estuary ecosystem, mangrove, rainforest, desert, all the different type of ecosystems, my dear children. This becomes a part of ecological diversity. So, what is genetic diversity? Variation within a species at the genetic level. Cool? Works? Now, going ahead, mention the evil quartet of biodiversity losses in a given habitat. What do you mean by evil quartet? Evil quartet refers to the 4 major causes of biodiversity depletion. Okay, 4 major causes of biodiversity depletion. Okay, you have habitat loss and fragmentation. For example, I am staying over here in the studio. Someone takes off the studio from me. Where will I go and stay? And slowly, if I do not have place to stay, I do not have place to have food, basically I will be dying off. You get it? So the most significant cause of biodiversity loss involving destruction or breaking apart the natural resources which are there. For what reason? For probably planting things, for probably crops. These things are going to lead toward biodiversity loss, my dear children. So first point will be habitat loss and fragmentation. Okay. Now the next point that we have is over Overexploitation. What is that? Now we have overexploitation. What do you mean by overexploitation? The excessive use of natural resources. For example, I have certain things. Okay, I have, for example, basically I'll tell you. So there are 5 biscuits. Again, I'm telling about biscuits. You might be thinking, ma'am is so hungry. Okay, so if there are 5 biscuits, my stomach would get filled with 2 biscuits. But still, I'm like, I should be eating those 5. And there is another person who is actually hungry. But I'm not giving, I'm a very bad person, okay? I'm taking all the biscuits over there. Am I not overexploiting? Yes. And what will happen? Basically, the biscuits will be finished up. It will be finished up and that guy would not be having anything to it. But that's not the problem. The problem is that the biscuits are getting finished up. There won't be anyone who can eat those biscuits now because everything is in my tummy. So excessive use of natural resources such as overfishing, poaching, logging, etc. leads to what? Population decline and extinction. A real-time example I'll give you. In Russia, there was Thalassemia cow. There was Thalassemia cow, which is an organism that you find in water. Now, this was excessively hunted. And when it was hunted, my dear children, after some point, it got extinct. So, it's a result of overexploitation. Aren't we doing a biodiversity loss over here? Yes, we are. Now, alien species invasion, that is also one thing. That is, for example, we think, oh, that particular plant is so beautiful. We'll take it to our country, that is India. When we take it to our country, that is India, the other organisms which are actually living there, you call them what? Indigenous species. The ones that are already over here, we call them indigenous species. Now, into that, we bring the invasive species, which will be fighting with our species for light, for food and everything. And slowly, what could happen is that these invasive species might wipe out our indigenous species. You get it? For example, water hyacinth. You might have studied it from your 10th grade or I don't know when, until when. Okay, so over here, water hyacinth or ecornia, what you call it, it spreads really rapidly. You can see, if you are in Bangalore, you can see that. Okay, and lots of lakes, there are so many ecornia being spread like that. Okay, now what do is that they do not allow the light to enter to the organisms which are down. The plants which are there deep inside, they do not get light. And also, when these ecornia and everything dies, there is too much of organic matter. Again, there will be algal bloom and a lot of things happening up there. There's a lot of problem happening up there, my dear children. So, introduction of non-native species into an ecosystem disrupts the balance and can outcompete or even eliminate the native species. I will give you more examples to this. It's a very important thing. Okay. Now, extinction, co-extinction. What do you mean by co-extinction? For example, I was dependent on you. Okay. Now, when you died, what did I do? I also died. So, this is co-extinction. Not in this manner. First of all, you should not be doing this. Okay. Now, listen to me. Co-extinction accident, for example, there was an insect and this insect was completely dependent on a plant for pollination, for laying its egg. Okay. And this plant was completely dependent on this insect for pollination. This insect species died. This plant species also will be dying because there is no one who could be doing the pollination. So, that also has to happen. When one species becomes extinct, dependent species may also, perish. So, habitat loss and fragmentation, overexploitation, alien species invasion and extinction, co-extinction are the 4 things that comes under evil quartet. Okay. Now, going ahead with the next thing. What do we have? Define the terms endemism and bioprospecting. Endemism and bioprospecting. What do you mean by that? Endemism means ecological state where a species is restricted to a specific geographical area. It is not found elsewhere, very unique and not found elsewhere. Example, lion-tailed macaque in the Western Ghats. You cannot find them elsewhere. This is known as endemism. Okay, where an organism is restricted to a specific geographical area and not found elsewhere. Lion-tailed macaque in Western Ghats. Very important examples, you should be mentioning it. Okay. Now, bioprospecting means the process of exploring biodiversity for new resources such as medicinal compounds, genes, biochemical products, everything which have what? Potential commercial use. The process of exploring what? Biodiversity. For what? For bioresources. For new resources, medicinal compounds, for genes, for biochemical products, all of this. That is known as bioprospecting. All these terms, no, standing crop, this and all, you tend to forget it. That's why I'll request you to have a notebook and write it down once. At least once you will remember it. Okay? Endemism, very unique. You are endemic. Okay, so basically not pandemic, you're endemic. That means you're restricted to a specific geographical area and are not found elsewhere. Now, what are sacred groups? Again, very important thing. What are sacred groups? For example, I am a person who has great religious beliefs, and which is why I have some religious association with the place. And I am saying that we should be safeguarding that place and things like that. But while I am doing so, I am also safeguarding the plants, the organisms which are up there. This is known as sacred groups. This is a form of in situ conservation. I am conserving organism in their original habitat. This is an example of in situ conservation, my dear children. So, forested areas protected by local communities due to religious and cultural beliefs, helping in the conservation of biodiversity. Examples are really important. You have Aravalli Hills in Rajasthan, Western Ghats region in Karnataka and Maharashtra, Sarguja, Chanda and Bastar of Madhya Pradesh, and in Meghalaya, what do you have? You have Khasi and Jaintia Hills. Don't write Khasi and Jaintia Hills, Meghalaya. Okay, Meghalaya is the place, and there you have Khasi and Jaintia Hills, very, very important. And I'll tell you, in Meghalaya, sacred groups plays a very, very important role. Very, very, very important role. Okay, especially in Meghalaya. They are like, safeguarding a lot of organisms just because of sacred groups. Okay, so Aravalli Hills of Rajasthan, okay, Western Ghats of Maharashtra and Karnataka, Sarguja, Chanda and Bastar regions of Madhya Pradesh, and in Meghalaya you have Khasi and Jaintia Hills. Okay, works? Now next thing. Among vertebrates, which group of animals has the highest number in global biodiversity? Vertebrates, as in the organism that has the vertebral column, my dear children. So, invertebrates comes fishes, invertebrates come, what do you call them, reptiles, amphibians, mammals, aves and everyone. So, among them, who has the most highest number in global biodiversity? It is fishes. It is what? Fishes. If you are asked about invertebrates, it will be insects. It will be insects. Okay, if you are asked about invertebrates, it is insects. If you are asked about vertebrates, it is fishes. Now, name the forest which is referred to as the lungs of the plant. It is the very famous Amazon rainforest, my dear children. It is known as the lungs of the plant. Okay, because it is having too many organisms, plants and all. It produces too much oxygen. Now, tropical rainforest has greater biodiversity compared to temperate forest. Why so? So, temperate region is colder region, and tropical regions, obviously, you know, there's a warmth in there, right? So, why do you think that tropical areas has more biodiversity than temperate regions? You only think in temperate regions there are chances for frequent glaciation. Whenever glaciation happens, what will happen? All the plants and animals will be dying off. So, that is a very big problem, right? So, wait, where is that point? Yeah, so longer evolutionary time. So, these forests have remained undisturbed, as in tropical forest, they remain undisturbed for a longer time, whereas what happens? Undisturbed by glaciation, whereas other temperate regions are disturbed by glaciations and everything. So, they do not allow time for evolving. So, they do not allow time for evolving, and evolution is not happening. That means biodiversity loss could be happening any time. So, they have longer evolutionary time. Apart from that, there is favorable climatic condition. You know, tropical rainforests have warm temperature, high humidity and everything, which will be supporting the survival and the growth of variety of species. High productivity also. If you have high productivity, obviously you can make sure that other organisms can also live over here. If you are producing more food, other organisms can also come over here and depend on your food. You get it? So, they have higher productivity. Why? Because they have ample amount of sunlight. Yeah, in tropical forest, you have ample amount of sunlight, my dear children, and also enough amount of rainfall. So, tropical rainforests have high primary productivity, supporting diverse food chains and complex ecosystem. I hope you are very clear with that. Yes? Now, going ahead, explain the species area relationship of biodiversity as explained by Alexander von Humboldt. So, Humboldt, okay. So, species area relationship, it says that Alexander says that basically the species richness increases as we increase the explored area. If we go ahead with the explored area, what will happen? The species richness will also keep on increasing. The relationship between species richness and area is expressed as S is equals to Ca to the power Z. What is S? S is the species richness. What is A? A is the area. Now, I will say, if I say the species richness is directly proportional to A, it won't be entirely correct. It would not be entirely correct, because after a point of time, it won't be increasing. That is what is put on over here. Species richness increases with an increase in explored area, but up to what? A certain limit. After that, it won't. So, even if I go up the area, the amount of species richness, it will be constant after some point of time. It will increase for some time. So, I say it's partially proportional. So, I have to change the proportionality constant. When I change the proportionality constant, I get C. And then I put A. okay. And since it is not completely proportional, I say that it is proportional up to a certain limit, and that limit is explained by z. Okay. Now, if you want to have a real-time understanding of things, you have to convert into log. So, in log, when you are doing, whenever you are calculating log, when you are multiplying log, you just put log s. Okay. When you are multiplying this, when you are multiplying log with this, my dear children, what happens? You put it as, you have to separate them, and you have to put something like this, log a. And whenever there is a power in log, my dear children, you have to write it in front. So, this becomes a thing, log s is equals to log c plus z log a, okay, where basically over here, yeah, I think that is quite understandable, right. So, for smaller values, z value ranges from 0 to 1, 0.1 to 0.2, and but for larger areas, the z value can be 0.6 to 1.2, indicating a steeper slope and greater biodiversity. So, z represents basically, as I told you, the amount of area that is being seen over here, an amount of biodiversity which is there, okay. So, this is a thing by Alexander von Humboldt, this equation and this line is very much important, just understand that. Now, wait, there is one thing that I was missing. Okay, here c represents the intercept of y, okay. So, wherever this line is going to touch y, that is will be the c intercept, or sorry, y intercept, and that is represented by c, cool. So, here everything is, c is a constant, that is the y intercept, and z is the slope of the line, regression coefficient, you call this what? Regression coefficient. Now, comment on David Tillman's long-term ecosystem experiments and its significance. What is David Tillman's experiment, my dear children? So, basically he had a setup, and according to his setup, he said that basically he is going to study the effect of biodiversity onto ecosystem stability and productivity. He has to see that. He had to see that whether if biodiversity is increased, does the stability and productivity also increase or does it decrease? So, what was his findings? His experiments demonstrated that higher biodiversity leads to greater productivity, more productivity, and also more ecological or ecosystem stability. Why, why dear children? Why do you think so? Because you have more organisms. So, for example, I have 3 organisms, and there are 3 organisms which are feeding on these organisms. There are high chances that they could just kill all of them and that's done. It's done and dusted. There is no stability. Where is the ecosystem? But you have so many options, my dear children. This will eat little of this, this will eat little of this, still there will be something left behind. So, the ecosystem will still be stable, and also there is higher productivity, that means more organisms can come and feed over here. So, significance, ecosystem with greater species richness showed less year-to-year variation in productivity and were more resistant to environmental differences like disturbances like droughts. Obviously, not all organisms can survive, right? So, if you have similar type of organism, all of them will die. If you have different type of organisms, if you have greater species richness, at least some of them would be surviving. Okay, that is the significance. So, the finding is this. The significance is that species richness would lead to what? Less year-to-year variation in productivity. Conclusion, these findings emphasize the importance of conserving biodiversity to maintain stable and sustainable ecosystem. And also one very important thing is that findings is that there was less wastage of resources. There was less wastage of resources because everything was being utilized properly of resources. Sunlight or whatever food, everything is there. So, higher diversity leads to greater productivity and ecosystem stability and also less wastage of the resources. This was David Tillman's experiment. Now, we have rivet-popper hypothesis, which was put down by Paul Ehrlich. What did he want to say with the help of this? So, basically, he thought of ecosystem as aeroplane. Ecosystem is what? Aeroplane. Now, in aeroplane, you have so many rivets. You have so many rivets. These rivets are the species. These rivets are the species. So, if I remove the rivets, what will happen? The aeroplane will go like this, right? It won't be stable. Now, suppose I am removing the rivets from the wings. Those are the most important things. And these rivets shows or is equal to what? The keystone species, the key species which are there for ecosystem maintenance. So, the rivets on the wings show the key species which are very much important. If I remove them, then the whole stability of the aeroplane, whole stability of the ecosystem also would be gone. So, rivet-popper hypothesis by Paul Ehrlich, and its significance is that he compared species in an ecosystem to rivets in an aeroplane wing. Just as losing a few rivets may not immediately cause a plane to crash, but losing a few species may not initially eventually affect an ecosystem. But what? Impact of species loss. If too many species, if so many rivets are being removed, initially you do not see anything because you are removing it slowly. But slowly, when you are removing a lot of them, the ecosystem may eventually collapse. Some species like key structural rivets are more crucial than others. Please remember that. Now the significance. This hypothesis highlights the interdependence of species. Yes. And warns that continuous biodiversity loss can lead to what? Ecosystem failure at the end. Conclusion. The rivet proper hypothesis stresses the need for biodiversity conservation to maintain the ecosystem stability and its function as well. Works? Works? Chalo. Next thing. Now, briefly explain narrowly utilitarian reason for conserving biodiversity. Now, you have different reasons for conserving biodiversity. One is narrowly utilitarian reason. What is narrowly utilitarian? Basically, we are getting certain products from the nature and hence we should be conserving biodiversity. We are so bad, right? We are so selfish. But yes, this is narrowly utilitarian point of view for conserving biodiversity. Now, direct economic benefits like human-derived food like cereals, pulses, fruits, firewood, fiber and lot of things. Okay. Apart from that, if you talk about industrial products, what do you have? You have tannins, lubricants, dyes, resins and perfumes. Yes. And then you have medicinal importance. So, over 25% of the global drugs are derived from the plants, my dear children, and 25,000 plant species contribute to what? Traditional medicine. Potential discoveries. Many medicinally valuable plants in tropical rainforests remain what? Unexplored. That is not needed. Okay. And bioprospecting is also been mentioned over here. Exploring genetic and species level diversity for economically important products. Okay. That is not important over here. These are the things that is important. At least three. Direct economic benefits that we get is cereal, pulses and everything. Industrial products also we get. Then medicinal things also we get. And that is how, that is why we should be conserving what? The biodiversity according to them. Okay. Now going ahead, briefly explain broadly utilitarian reasons for conserving biodiversity. Now according to this, you should be conserving biodiversity for the services, ecosystem services they provide. For the ecosystem services they provide, as in good amount of oxygen, okay, pollination and everything. Because if pollination is happening, that at the end you will get the fruit, right? So get it, flowers, a lot of things. So oxygen production, the Amazon rain, over here you are talking about the products. Over here you are talking about the services. Okay, the Amazon rainforest produces 20% of the earth's oxygen through what? Photosynthesis. That is one thing. Now pollination services, bees, bumblebees, birds and bats help in pollination, which is essential for fruit and seed production. Now economic value of ecosystem services. The cost of oxygen cylinders or artificial pollination highlights the economic importance of nature's free services. I told you, right? And fruit and seed production is done, oxygen is done. Okay, and now aesthetic and psychological benefits. Biodiversity provides intangible pleasures like walking in the forest, enjoying blooming flowers and everything. That is something that you cannot compare, right? So these are the reasons why you should be conserving biodiversity in broadly utilitarian point of view. Now going ahead with the next question, what is India's share in global species diversity? What is it? It is 8.1. It is what? Yeah, it is 8.1 of the global species diversity. Now, how many mega diversity countries are present in the world? How many mega diversity countries are there? There are 12 mega diversity countries in the world. 12. Please remember, I told you these technical terms are really important, okay? Now, why Western Ghats range is considered as one of the best biodiversity hotspots, my dear children? Why do you think it's a hotspot? Because it has high species richness, and also you find endemic species where you cannot find anywhere else, such as lion-tailed macaque, right? Now, home to several endangered and rare species of flora as well as fauna, as in plants as well as animals. It has diverse ecosystem including tropical rainforest and montane forest as well. Now, faces threats like deforestation and habitat destruction, qualifying it as a biodiversity hotspot. So it has high species richness and they have endemic species. It is home to several endangered and rare species. It has a very diverse ecosystem as well. This is why you consider it as a biodiversity hotspot. Going ahead, Name any 4 recent extinct organisms as per IUCN list. Very important question. You have dodo from Mauritius, quack from Africa, thylacine from Australia, Thalassemia from Russia, and 3 subspecies of tiger, that is Bali, Java and Caspian. I will request you to go see their pictures, images. In that way, you'll remember it better. So, dodo, quack, thylacine, Thalassemia and 3 subspecies of tiger, Bali, Java and Caspian. Works? Now, what is ex situ conservation? Mention 2 example. Ex situ conservation as in, I am conserving organism outside their actual habitat. Outside their actual habitat. For example, zoos. For example, botanical gardens. Okay, for example, I have seed banks. I have the seed, but it's not from there, right? I'm conserving it somewhere else. Okay, for example, wildlife safari parks. Wildlife sanctuary is different. Wildlife safari park is different. Please remember that. So, and also conservation of gamete by cryopreservation. So, ex situ conservation says threatened plants and animals are taken out from their natural habitat, not because we want them, but that's how they can be conserved. Okay, because it's risky to keep them there, and placed in special setting where they can be protected and given special care, or protecting threatened species of plant and animal outside its natural habitat under what? Human care. Example, zoological park, that is zoo, botanical gardens, wildlife safari parks, and conservation of gametes by cryopreservation, or what do you call? Then you have seed bank as well. Okay. Now, next question. Alien species invasion caused decline or extinction of indigenous species. Justify the statement. I told you this is very important. Where you're bringing things from outside and they're going to cause trouble for indigenous species, it is very much important to discuss about that over here. So, first thing that you have is Nile perch. Nile perch is a very big fish. You know, Nile perch was introduced into Lake Victoria in Africa. And what happened? There were so many cyclid fishes. What happened to these cyclid fishes? They all kind of got extinct because of this Nile perch. Okay, because when Nile perch was introduced into Lake Victoria, it led eventually to the extension of ecologically unique assemblage of more than 200 species of the native, it's native African cyclid fishes in the lake. Apart from that, you have African catfish. It's happening in Africa only. Okay, it's African catfish, that is Clarias garipinus. Clarias garipinus. This is, guys, very bad. It was used for aquaculture purposes, but it actually posed threat to for indigenous catfishes in our rivers. African catfish, Clarias garipinus, in our river, they were causing trouble for indigenous catfishes. Very bad, right? Yeah. And now you have one more thing, that is for plants. Okay, for plants, you have carrot grass, that is Parthenium, lantana, and also water hyacinth, which is known as Eichhornia. All of them basically are the invasive plant species. Carrot grass, that is Parthenium, lantana, and water hyacinth. Please remember the names. Please, it is very important. Okay, if you're getting direct questions, just score good marks in direct questions. That will be better. Fine. Now, are we done? Are we actually done? Oh my God. Okay, I didn't expect that coming. So, my dear children, make sure, since it is already done, let me tell you, Toto Bashan, okay? So, there are questions that you might have not understood. I'll request you to revisit those questions or the concepts, okay? Please go through it once more, settle in your head, and you will be able to remember it better. Now, I have given you a strategy to follow. Please follow that. Practice the important diagrams. Diagrams are also equally important. Okay. In the strategy video, I have told you what all diagrams are important. You can practice those diagrams. Works? When you solve all these questions, you might feel a sense of cessation. That is, I cannot do it anymore. It's okay to feel that way. It's totally fine. As a student, I can also relate to you. Okay. I can also think, even I am studying right now. I also get bored. I cannot take it anymore. I also get you. But you know what? This has to be done for just few more days. Just few more days and then it's done. So please think in that sense and go ahead with your preparation, children. You are going to ace your examination. Trust me on that. Wishing you all the best. If you need any of my help, do let me know in the comment section. I am here to help you. Lots of love from Jasmine ma'am. Take care of yourself. You are going to do so good.